ahead and start recording now. Yes. Okay, so we're recording. All right, so a couple of people jumped on a bit later, but we've just had a really nice introduction to SEGH by our president, Michael Watts, located over in the UK. So this is sort of how we're going to move ahead going forward. So I've already talked to a few of you. Basically, I'm going to sort of work on the front end and be, sorry, the scruffy looking face of the conference. We have Alistair, who has all the presentations loaded up and ready to go. So unfortunately, we couldn't let everybody share their own screens just because we would never get everything done sort of on time. So Alistair is going to be running them, and he'll simply pull them up. And then when you want to go to your next screen, just say next. So we'll say all of our please and thank yous up front. And if we just say next, then we can kind of go through there. So I'll be monitoring sort of time, and I'll let everybody know. So for 15-minute speakers, I'll let you know when you have about 3 minutes left. And for the 5-minute speakers, I'll let you know when you have like 30 seconds left, just so you can wrap up. And I'll try to be a little bit more lenient on maybe the PhD students and sort of earlier career people on time, but we'll try to keep everything in time, at least within the different sessions. So session 1 and session 2. Okay, so with that in mind, I'm going to give the floor over to Bob, and he has a few extra seconds. So take it away, Bob. And we're really glad to have Bob. He's semi-retired now, but he's been part of SEGH for a long time in the Americas. He knows about what's going on, and he still has a lot of energy, and he's sort of leading our charge of trying to rebuild our numbers. So thanks, Bob. Take it away.
Thank you, Clay. I'm between retirements. Good morning, everyone, or good afternoon, as the case might be. Apparently, I am the last man standing. I've been a member of SEGH since 1990 and may be the longest serving active member in the Americas. And therefore, I've been asked to give a brief overview of the history of the organization in the Americas. To do this, I am relying heavily on a recent memo prepared by one of the earliest members of SEGH, Dr. Brian E. Davies. In his memo, Brian noted that the need for a Society for Environmental Geochemistry and Health arose in 1968 at a symposium on environmental geochemistry and health and disease that was held in Dallas, Texas, coincidentally, just a few miles south of where I am now. The SEGH came into formal existence at the 4th Annual Conference on Trace Substances in Environmental Health. And that was in 1970, with 11 founding members, making it one of the oldest specialty societies. One of the founding members was the late Harry V. Warren from Canada, who in 1970 invited Professor Davies to join SEGH. Thus, he became one of the earliest members of the society. Brian has maintained his membership ever since and may be one of only 2 surviving members from 1970, the other being Bobby Wixon, who at that time was with the Missouri School of Mines in Rolla, Missouri. Now, much of the early research and SEGH leadership came from this university, where Brian Davies was also a visiting professor. Davies was elected as counselor of SEGH in 1981 and was elected president for 1989 to 1991. Davies writes that in the 1980s, university and research institutes started to experience increasing funding problems. In Britain at that time, there was approximately a dozen SEGH members who had been faithfully attending the Missouri conferences. But the increasing shortage of travel funds made continuing attendance in American SEGH conferences problematic. An idea emerged that in order to retain the British membership, European meetings should be organized as part of SEGH outreach. Unfortunately, with the retirement of Bobby Wixom and Brian Davies from active research, the focus of SEGH shifted from the Americas to the UK and Europe. Incidentally, also in 1985, Brian Davies was asked to be the founding editor for a new journal, Environmental Geochemistry and Health, and that's still very much active. These are critical times. We are all aware that COVID-19 has killed more than 700,000 people in North America and almost 5 million people worldwide. But few people realize that the natural environment kills that number each and every year. And that's not even considering the health impacts of anthropogenic pollution and the impending impacts of climate change. We now have the opportunity to reinvigorate SEGH Americas with bright, enthusiastic young researchers, a number of whom are in attendance in this meeting right now. We all have important roles to play in addressing these environmental health issues, and SEGH Americas can once again be the leader in this effort. I also want to bring your attention to another conference that SEGH is co-sponsoring. It's a student-oriented conference on environmental health and medical geology. This year's focus will be on Latin America. The meeting will be October 18th and 19th. The deadline for submitting abstracts is the end of this month. If anyone is interested, everyone is welcome. Please contact me. My email address is bobf@utdallas.edu. I thank you for your attention and turn the floor over to Clay.
Clay, can I just add for the details of the meeting are on the SEGH website, segh.net.
Thanks, Michael. Yeah, and thanks, Bob, for that really wonderful introduction. And I think, yeah, now's the time where we're all, acutely aware of health. So I think that, I'm really excited about this meeting. So with that, we'll bring up our first speaker. So we have Malcolm SEGHL. And he's really big in this movement, and we're really glad to have him here. So he'll be delivering his talk called Medical Geologists on the Team, New Roles in Exposure Science and Risk Management. So whenever you're ready.
Yeah, thanks. So Alistair, I'm going to be sharing my screen, doing it from here. So let's get that started there. And okay, does everybody see my?
That looks good.
Okay, great. Okay. And I'm just going to minimize one more thing. Okay, good. Well, good afternoon. My name is Malcolm SEGHL, and I'm the current chair of the Geology and Health Scientific Division of the Geological Society of America. I've identified myself as a medical geologist for the last 15 years, yet I'm still grappling with the question, what is medical geology? And where do we fit into the broader fields of the geological sciences and public health? So today I'd like to provide some ideas on where the training we get as geologists or public health scientists can be used in the context of risk assessment and exposure science. First, I'll talk about risk assessment within the context of the environmental pathways and biological impact model. And I'll describe two concepts that I think are useful in medical geology and risk assessment, namely the geoavailability and the bioavailability. Then I'll talk about how medical geology and exposure science are great partners. I'll talk about some new concepts in exposure assessment and tools such as omics technologies. I'll briefly describe the unifying concepts in monitored exposure science, the exposome and the meet in the middle approach, where I think our geological and biomedical expertise come together. Source to outcome models are used in the risk assessment to link the discharge of a contaminant into the environment and resultant health effects in a population. The environmental pathways and biological impact analysis starts with the discharge of a contaminant into the environment, where its concentration and speciation are controlled by hydrogeochemical and biogeochemical processes such as dilution, chemical reaction, and physical transport. Once the contaminant is in the environment, people can be exposed, and their exposure is affected by their activity patterns, including occupational exposure and demographics. This exposure leads to a biologically significant dose, which then results in health effects which are influenced by the responsiveness or susceptibility of the population. The geoavailability describes the fraction or amount of the contaminant in the environment that's actually available to reach a potentially exposed population. Not all of the material that reaches an exposed population will actually have a health effect. This requires assessment of the bioaccessibility relating exposure and dose. Next, the bioavailability relates to the amount of toxicant that reaches a biologically important site in the human body and leads to health effects as described by a dose-response relationship. So first, I'll focus on the environmental pathway part of this analysis, where we describe the geoavailability. The tools for environmental pathways analysis are familiar to most earth scientists. The tools of chemicals and materials and engineering and science are used to characterize the discharge of the pollutant from the source. The skills from geochemistry and hydrology are used to describe the processes that affect the pollutant in the environment. And then we characterize its presence in the environment using the tools of environmental monitoring, geostatistics, and GIS. The result of the analysis at this point is the estimate or description of its geoavailability. The geoavailability can be reduced by a number of biogeochemical processes which remove the contaminant from the mobile phase, rendering it unavailable for further transport. A number of standard geochemical techniques have been used to estimate a qualitative geoavailability. These include sequential extractions to identify the host phase for a contaminant, and electron microprobe analysis, which show elemental associations that suggest the host mineral phase. We can calculate a quantitative geoavailability factor using well-accepted principles of contaminant transport. Consider a representative elementary volume, the REV, for contaminant transport calculations in a porous media with porosity theta. The concentration of the contaminant in the fluid is C sub L. The concentration of the contaminant in the solid is C sub S, and the solid density is rho S. Then the amount of contaminant in the fluid is C sub L times theta. The amount of contaminant associated with a solid is the product of the terms involving concentration, porosity, and solids density. The total amount of the contaminant in the unit volume is their sum, and the fraction of the contaminant in the fluid that can be transported to a potentially exposed population is shown by this final expression, which I'll call the geoavailability factor. We can also calculate the geoavailability factor from simple terms in contaminant transport equations. The retardation factor, R, compares the travel time or the travel velocity of a contaminant to a reference, usually the fluid flow. The standard equation for retardation factor in porous media involves K sub D, the distribution coefficient, which is the ratio of the contaminant concentration in the solid divided by the concentration in the liquid. If we substitute those terms for KD into the retardation factor equation and collect terms, we obtain the ratio of the total amount of the contaminant divided by the amount in the liquid phase. Taking the reciprocal, we see that the geoavailability factor is simply 1 over R. There are a number of methods to estimate the retardation factor and the geoavailability factor. These include batch sorption and effective porosity measurements, which yield KDs, column transport experiments, which provide direct measures of the retardation factor, in situ retardation factor using analysis of groundwaters and solid samples taken in the field, and finally, reactive transport modeling. Now let's turn to the biological impact analysis of the risk assessment framework. Personal exposures are measured by microenvironment monitors, wearable personal monitors, and internal monitors. The relationship between exposure, dose, and health effects are studied with the tools of toxicology and studies of the metabolism and the mode of action of contaminants that are responsible for the dose-response relationship observed. Whether a population will experience significant health effects from a dose is very dependent on other characteristics, such as its general health and genetic makeup, which can be studied by the tools of molecular epidemiology. The relationships between exposure, dose, and health effects are impacted by the bioaccessibility and the bioavailability. In medical geology, we want to understand the linkage between the external environment and internal exposure. We can describe exposure pathways and exposure routes responsible for external exposure, and biomarkers that can describe internal exposure or dose. Exposure pathways are the media that carry the contaminants. Exposure routes include ingestion, dermal absorption, and inhalation. Finally, biomarkers are indicators of either exposure or disease and are obtained in biological samples such as blood, urine, and hair. We can distinguish between biomarkers of exposure and biomarkers of effects. Biomarkers of exposure relate external exposures to internal exposure and include measurement of metals and biochemical complexes in bodily fluids and tissues. We define the bioaccessibility of a contaminant that's been ingested as a measure of the fractional dissolution of the contaminant from an environmental media into a biological fluid. The bioavailability describes the fraction of the chemical that is absorbed into systemic circulation. The concentrations and speciation of these biomarkers are determined by chemical and physical processes within different body systems. So as we follow the fate of a contaminant absorbed onto a geological matrix, we see that in the digestive system, only a fraction of the total contaminant is released by pH-dependent dissolution or desorption, and then a fraction of that is absorbed by the small intestine, and then a fraction of that passes through the liver without being metabolized and reaches systemic circulation and its target. The bioavailability is the product of these fractions. The bioaccessibility will be influenced by many of the same geochemical features and processes as the geoavailability. It'll be sensitive to the mineralogy of the soil, the solution composition, and also to the particle size and nature of the mineral coatings on those particles. Processes such as precipitation, adsorption, and desorption will control the amount of contaminant that's available for subsequent absorption and metabolism. Standard geochemical tools can be used in bioaccessibility studies. However, there are some differences to consider, such as additional chemical components, such as amino acids, proteins, and enzymes. The residence time of small particles in the gut is different than geologic residence time, and chemical conditions change rapidly as particles move through the GI tract. Biomarkers of effects are used to recognize preclinical manifestations of diseases and include a large number of molecular biomarkers and changes in cellular function and morphology. Some of the biomarkers or of effects for your arsenic and uranium are listed in this table. Micronuclei can be used to assess DNA damage for a variety of potential cancers. The COMET assay is used to directly measure DNA damage. Under the influence of an electric field, damaged strands of DNA lose their compact structure and are separated from undamaged strands. The resulting pattern resembles a comet, with the head containing undamaged DNA and the tail containing fragments of damaged DNA. The most complex set of biomarkers include those profiled using omics technologies, such as epigenomics. Omics deals with the characterization of groups of biological molecules that control the structure, function, and dynamics of an organism. These include genomic studies in which the DNA in people who have and do not have a disease or condition of interest are compared. Epigenomics, the analysis of chemicals that regulate gene expression or function. Adaptonomics, the analysis of chemicals that bind to DNA or selected proteins and interfere with the DNA function or replication. And transcriptomics, the analysis of variations in RNA that prevents the process of transcription and synthesis of proteins. This leads us to the next concept for this talk, the exposome. In the last decade, genome-wide association studies failed to explain most variability in human diseases, and more interest in environmental factors has emerged. In 2005, Christopher Wilde defined the exposome as the totality of a person's exposure from conception to death as an analog to the genome. In addition to differentiating between specific external and internal exposures, this recognized the importance of the general external environment as a determinant of human health. This general external environment includes things like social capital, education, psychological stress, and climate. The omics technologies and environmental technologies I described earlier are integral parts of exposome research.
3 minutes, Malcolm.
One of the important tools in exposure science is the use of physiologically based pharmacokinetic models. In these models, different organs are represented as compartments. Transfer between compartments is characterized by a flux. This kind of modeling should be familiar to geochemists who have modeled Earth systems for decades. The diagram on the left is from a book on geochemical cycles by Bob Garrels in the mid-1970s. The PBPK model to the right shows a feedback loop between the liver, bile, and blood. The pharmacokinetic models can be run in the forward direction, that is starting with external exposures and how they lead to internal exposures and health effects. Alternatively, these models can be run in the reverse direction using measured concentrations of chemicals in different body fluids and then working backwards to infer external exposure. This leads to the final concepts that I want to introduce, the bottom-up and the top-down approaches and the meet-in-the-middle concept in exposure science. In the bottom-up approach, research is exposed on each category of external exposure to find biomarkers of exposure to characterize individual exposomes. The second complementary approach, the top-down approach, focuses on biomarkers of effects, uses untargeted omics methods to characterize the different chemical markers in a single sample of biological fluid. Then the omic profiles of diseased and healthy individuals are compared, and then hypotheses are generated to identify particular exposures. Each of these approaches is characterized by significant uncertainties. However, this meet-in-the-middle approach leads to reduction in uncertainty when they arrive at similar conclusions. Finally, we can return to the source-to-outcome model and show how different methods can be used at various stages of a risk analysis. Different methods are useful for describing exposures for different time periods, different spatial scales, and require different resources, but together they can be used to describe major parts of the exposome. For example, general external exposures are often evaluated through questionnaires or by mining socioeconomic databases. Geological insights can give information about likely external exposures over long or short periods of times. Specific external exposures are measured by environmental measurements of air, water, and soils, as well as personal monitors coupled with GIS and GPS. Internal exposure is measured by the biomarkers, such as the omics technologies mentioned before. The purpose of my talk was to acquaint you with some of the tools being used in the biomedical sciences that may be unfamiliar to medical geologists. This may help you become more effective in teams involving medical and geological professionals in the public health sector. In closing, I want to mention 2 new resources for learning about these topics. The book Practical Applications of Medical Geology has just been published by Springer, and at this year's GSA meeting, the Geology and Health Division is sponsoring a session on these topics. And I thank you for your attention.
Thank you, Malcolm. Thanks for the great overview. And we'll have a little bit time of questions after this first section of talks. We can go in there. So if Alistair will pull up the second talk, we have David Patch talking with a title, presentation entitled From Sock to Stream. From Sock to Stream, Environmental Release, Transport Fate, and the Effects of Weathered Silver Nanomaterials from Commercial Products. So take it away, David.
Thanks so much, Clay. And just to confirm, I say next, and then the next slide will go?
Yeah, for sure, yeah.
Yep, okay, sounds great. So hello everyone, welcome. Thank you for having me. And before I get into my presentation, I want to pose the following hypothetical situation to you all. Imagine you're in the lab looking through a microscope day in, day out for weeks, when finally you realize you've done it. You've discovered the cure for everything. So you turn to your colleague, raise your arm for a huge high five, but wait, you haven't showered in weeks. Next slide. And now you're worried you just caused a huge lab incident by basically stinking out all of your colleagues. But you smell yourself and you're fine. Next slide. Because you realize that you have silver in your clothing, and silver is an antibacterial which kills off odor-causing bacteria, leaving you and your clothes fresh. So you go home, next slide, throw your clothes in the laundry and go to sleep knowing that you've just solved everything going wrong with the world. But what happens to that silver in your clothing when you wash it? Does that silver get released and make its way into the environment? And if it does it have an impact on soil and water systems all throughout the world? Well, next slide. Those are some of the questions we sought to answer through a multi-collaborative project, which I'll be presenting the first part of here today. So once again, thank you everyone for taking time out of your day to take part in this presentation. I'll be presenting my research looking at from sock to stream, characterization of released silver nanomaterials from commercial products after simulated wearing. I'd like to also thank Natalia O'Connor for taking the time to help with this project and look over my presentations, as well as Dr. Iris Koch and Dr. Kayla Weber for being my two fantastic supervisors and guiding me through this research. Next slide. So before I go too far into it, what exactly are silver nanomaterials? Well, silver nanomaterials are typically less than 100 nanometers in size, and they have enhanced surface area to volume ratios that give them unique properties compared to their bulk metal counterparts. Nanomaterials themselves come in a variety of different sizes, shapes, coatings, and speciations. And the fate, transport, toxicity of these nanomaterials is highly influenced by their characteristics. In the top left image, we have some spherical silver nanoparticles that you can see are roughly 100 nanometers in size. These nanomaterials can interact with and disrupt bacterial functions, which is why silver nanomaterials in particular act as antibacterial and to a lesser extent, antiviral agents. Depending on the shape and size of these silver nanomaterials, they can also exhibit different optical properties. You can see in the image in the bottom, we have silver spheres of varying sizes, which have different colors, as well as silver plates of different sizes, which also have different colors. Next slide, please. When the silver is potentially released from commercial textiles or other commercial products, it has the potential to transport and make its way into the environment. And as I said, this was a huge project which involved numerous grad students over a long multi-year study. I will be looking at the release of silver and talking about that here today, but we tracked the release of silver into constructed wetland systems as well as into soil systems to measure the overall impact in these different areas. However, if you're going to measure the impact of silver in wetlands and soils, you have to be able to really characterize what materials you're working with. One of our predictions was that silver nanomaterials that are being released from commercial products might be very different than those that you can go and purchase from a chemical supplier store. Next slide. So as I said, it'll be going over some of my research. Next slide. So the overall goal for the project can be easily broken down into three different areas. The first step was to develop a simulated human weathering method to generate representative weathered silver nanomaterials. The next step was to then fully characterize those weathered nanomaterials. And the last step was to investigate the impact of weathered nanomaterials compared to well-studied forms of silver, ionic and PVP coated. And as I mentioned, next click please for the animation. I'll be focusing on these 2 points here today, developing and then characterizing the silver nanomaterials. So how exactly do we mimic human weathering of a textile? Well, if everyone takes 2 seconds to think about it, think of, when you go for a run or go for a jog or go to the gym, what are the main stresses on your textiles? We identified the two main stresses as chemical weathering or sweat and physical weathering. So next slide, please. To start with chemical weathering, fortunately, there's a lot of recipes or methods in the literature to simulate human sweat. We took one from method ISO 105E, which is simply just a mixture of sodium chloride, sodium dihydrogen-orthophosphate, histidine, and adjust to pH 5.5 with sodium hydroxide. If we were subjecting the textiles to chemical weathering, we would basically subject them to this sweat for 60 minutes, in which we would basically take the sweat with a pipette and load it onto the textiles and let it basically sit there and soak. Next slide, please. To physically weather textiles is a little bit different, and this is something that no one's really done before in the literature. Now, we thought maybe we could just recruit a whole bunch of grad students, make them run around in circles for a bunch, but we couldn't really get the ethics approval for that. So instead, we decided we had to 3D print something. So we 3D printed here, 3D printed foot, one of which I have right in front of me right here. And we basically put it inside of a shoe with a sock on it and had it go for a run and a walk. And what this does is it simulates both abrasion, so the sock moving on a shoe, going back and forth, as well as the sock being stretched and being forced to kind of elongate around the foot itself. We also tested a physical weathering apparatus, which simply just stretched the textile itself. Next slide, please. So now that we've looked at how to chemically and physically weather a textile, we want to examine the impacts that those weathering techniques have on the amount of silver released from the textile, the figure on the left, and the speciation of that released silver, the figure on the right. So in the figure on the left right here, we have the silver released in milligrams of silver per liter of wash water. And on the right, we have the speciation of the silver released, looking at 4 different types of speciation. Focusing on the left graph first, we saw here that our NS control, or our no sweat control, resulted in a release of 2 milligrams of silver per liter of wash water. This is very comparable to what is often found in literature from other studies using very similar textiles, about 2 milligrams per liter of silver. When we add one form of when we add one form of weathering, either physical or chemical weathering, we see the amount of silver double from 2 to 4 milligrams per liter by either stretching, walking, or sweating the textile. When we combine that physical and chemical weathering together, we see the silver amount jump further to 6 milligrams of silver per liter of wash water. And then we took this and compared this to results that we actually generated from human trials, as was performed and published by a postdoc in our group, Dr. Vincent Gagnon. And he found about 4.8 milligrams of silver being released. The other key thing is the type of silver being released. In our chemical weathering trials, we saw a very, very tiny amount of ionic silver being released compared to the physical weathering trials where much more ionic silver was being released. Ionic silver is classically thought to be the most toxic form of silver. So by matching both the total amount of silver being released as well as the amount of ionic silver being released, we felt that the sweat stretch and sweat walk trials resulted in silver release characteristically similar to that of the human trials. We can see that replicated further when we look at our speciation results. We can see once again, just physical weathering or the control textile alone, we have mainly metallic or nanoparticulate silver being released in blue. As soon as you start to incorporate sweat, whether artificial in the next 3 trials or actual human sweat in the trial on the right, the human release, we see substantial amounts of silver being released as both silver chloride and metallic silver. Next slide, please. Now that we know what kind of silver is being released, we should probably try and figure out where the silver is coming from. We know it's coming from the socks, but what is it from the socks that is releasing silver? Is it dried silver salts? Is it some kind of silver wire? And what we found is that it's actually a silver plated nylon fiber. This is called the ecstatic nylon fiber. As you can see by these 3 little fibers in the middle, it is a nylon undercore that has been electrolytically coated in metallic silver. So once we identified the source of our silver, it's a thin nano layer of silver on this fiber, we wanted to then ask ourselves, what is physical and chemical weathering doing to the fiber and doing to the coating on that fiber? Next slide, please. So we compared a number of different fibers to each other, but for today's talk, we'll be just looking at the artificially weathered textile and the human weathered textile. And as you can see here, unlike the previous photo in which we saw a very nice, pristine, smooth silver surface, we see massive physical degradation to the surface. We see silver just missing in massive chunks. And we see some of the silver actually being pulled off or flaked off or kind of just chunked away from the surface. Also, we see the surface has undergone chlorination. The sodium chloride present in the sweat has resulted in a surface chlorination of the textile material. And we can see very similar images and results from our artificially weathered textiles to our human weathered textiles, suggesting once again that our simulated human weathering method is getting pretty close to the real thing. But we care most about what is making its way off of the textile. Next slide, please. So here we have 3 different sections of nanomaterial images taken from 3 different sets of wash water. With A, we have our control wash water, which has been subjected to no physical or chemical weathering. We see a large silver nano plate in the middle, which is actually about 10 micrometers long and wide, but only about 80 or 90 nanometers thick. And we see smaller silver particles spread all throughout. In red, we have our silver EDX speciation, and in blue, we have our chloride speciation. As you can see here in our A trial, we see substantial silver spread throughout the SEM image, but we don't see a lot of chlorine, if any at all. However, when we take a look at B, which is our chemical and physical simulated weathering, we see both substantial silver being released, as well as most of that silver being silver chloride, confirming our previous findings with our speciation results. And lastly, looking at our final row, our row C, which is our human weathered textiles, we see once again mass amounts of silver being released that consists of both silver and chlorine. Next slide, please. So as you can imagine, this study looked at a number of different variables across all the different types of weathering treatments, and it was a massive study that resulted in the generation of a lot of data. To summarize that data here right now, you can see the total amount of silver released across the trials, the amount of ionic silver released, the coating of the textile, and the speciation of the released silver. Next animation, please. And as you can see here, our physical and chemical combined experiments resulted in data and results that most closely match that of the human released experiments, suggesting that we actually have been able to generate or simulate human weathering of these textiles to mimic what humans do when they're using these textiles themselves. Next slide, please.
3 minutes. Okay, thanks. Yeah.
So in summary, physical and chemical stress influence the characteristics of released silver nanomaterials. Numerous analytical techniques are required to fully characterize released silver nanomaterials. This study, as I said, has resulted in the successful development of a simulated human weathering method. And these results and the findings suggest that human use of silver-containing textiles results in chlorination of released silver nanomaterials. Next slide, please. This was a massive study with a number of different collaborators. I'd like to thank Sophie Maynard, Kendra Buchholz, Sarah Denford, Chris Folkens, Marika Hasbeck, Christina Jones, Dr. Mark Button, Dr. Jennifer Snellgrove, and Dr. Vincent Gagnon for all their contributions and work to this study, as well as all the collaborators and institutions you see here. And lastly, I'd like to thank you, the audience, for taking time out of your day to take part in this presentation. Thank you so very much, and I'd be happy to take any questions.
Thanks, David. That was really great. And sorry, I kind of jammed you up at the end. Yeah, thanks.
No,
thanks
you're
for
good.
staying
You're
on
good.
time.
Please do.
If anyone has a quick question while we're pulling up the next talk, go ahead and just unmute and ask it. Or you can also put them in the chat throughout this whole process. So. Okay, I have one. I'm going to save it for the question period after. I think we'll go on, and then thanks for getting us back on time, David. I really appreciate it. That was great. Thanks.
My pleasure. All part of the plan.
Yeah. So for the next talk, we have Iris Koch, who was mentioned in that previous talk, giving a talk entitled "From Health Concerns to Data Quality: Investigations into an Underperforming Bioaccessibility Standard Reference Material." So take it away, Iris.
Thanks, Clay. It's nice to meet you after our little discussion yesterday.
Nice to meet you too.
I made it, yay.
Great.
Yeah, and nice to see everybody too. I'm showing my face here so that you know what I look like. So yeah, thank you for that introduction. Thank you also to you and to Alistair for inviting me to give a talk here. Hopefully it'll be interesting for people. I'd also like to thank the first speaker, Dr. Siegel, who gave a really good background to a lot of the things that I will be just touching on really, really briefly. So it was nice to have that so that I don't have to explain too much. So before I get started, so Alistair, I do have some animations, but I'll just probably say, click 3 times or whatever. And yeah, also I'm sure you can read my mind. Yeah. So yeah, of course, besides myself, I'd like to point out David, who just gave a talk, and Natalia O'Connor as all the people who contributed to this talk and co-authors, and also Alistair Kirolf, who I co-supervised with Diane Boscheman at Queen's University. And a shout out to Alistair, who just submitted his thesis today. He's finished, he's now Dr. Kirolf. So congratulations, Alistair. So next slide. So what I wanted to do here to sort of bring the health aspect into really sort of a QC conundrum that we have is to start out by telling a story. My introduction is going to be a sort of a story. So this is a purely fictional story, but we'll say it's about me and my family so that I don't embarrass anybody. And it starts with some contaminated soil or what we suspect to be contaminated soil. So let's say I live in a residential neighborhood, and this is a neighborhood that has had some historical contamination. And my neighbors and I have gotten together and we've expressed our concerns to, and actually I show there my garden because I have a garden and my neighbors have gardens, and we have this soil that we grow our gardens in, and we have families who go outside in our yards. And so we've expressed this concern to the local government, and the government has decided they're going to do a study to look at. They've hired a consultant to do a study to look at this issue. So next, Alistair. And so what they do is the consultant's going to come out and take some soil samples. It's not going to be a surprise to anyone here, and get it analyzed. And sure enough, we have found some contamination, and it's lead and arsenic, for example. So next. And the consultant doesn't just stop at identifying the contamination. The consultant's now going to do a risk assessment. So this is the process of essentially looking at the health concerns and health or the probability of adverse effects, and predicting that with more accuracy for our particular situation. So that involves looking at all the people who are involved, the age groups, and of course, like I said, we have families, we have children of all ages in the neighborhood, identifying the chemical hazards, which, as I've said, are lead and arsenic, and then the connection between the two, which is basically things like growing our gardens and also children playing in the soil. So next. And the outcome of the risk assessment is that toddlers are at risk from arsenic and lead. Here I have shown a picture of a toddler who happens to be my daughter when she was about 4 years old. And that was after she had taken a container of muddy water and tipped it over her head. So it's really just to show that we know that toddlers come into contact with dirt all the time. So next slide. And now the risk assessor has basically said, well, there's this way of looking at risk, and the risk is directly proportional to these things. And one of them is the contaminant concentration, not surprising there. Exposure factors, which is basically how much soil toddlers eat. And then that's divided by the body weight. And all of that is multiplied by this notion of bioavailability, which was explained to us earlier. So if you click 3 times, please. Thank you. Then we have, so this basically shows a very simplified definition of the bioavailability in the context of a risk assessment, but basically the amount absorbed that can cause the adverse effects. And the consultant has some reason to believe that where usually the bioavailability is assumed to be 1, so that means that everything that a toddler would eat has the potential, or all the contaminant present in the soil has the potential to be absorbed and cause adverse effects. The consultant has reason to believe that bioavailability is in fact less than 1. And that is because of the what they suspect to be the mineralogical forms of the arsenic and the lead present, that they are in less soluble forms. So the consultant now has turned to bioaccessibility. Next slide. So this is where I'm going to put my sort of project manager hat on, and this is basically to tell you that we actually I project manage all the various bioaccessibility testing projects that we have in our group. We actually provide this bioaccessibility testing to people in Canada, and we use the US EPA method, which I'm sure the people in the States are well familiar with. This is the in vitro bioaccessibility assay for lead and arsenic in soil. This is more or less recommended by Health Canada guidance, and we like it, and Health Canada likes it, and US EPA likes it, because this bioaccessibility method, which is a lab method, gives results that are representative of relative bioavailability results. And these are obtained through animal testing, which is, as you can imagine, very resource intensive. So when we have a method that gives us results that we think are pretty much the same as what we would get from animal testing, then that's a good method to use. So the method is basically, in this case, it is only the gastric phase that is represented. So it's a low pH, which represents fasting conditions. It's at 37 degrees, which is body temperature, with end over end mixing. Next slide. Now, one of the things that's really important that I rarely hear people talk about, but as my students know, is very important to me and something I kind of harp on a lot, is QA/QC, quality assurance, which is the program, and quality control, which are the specific tests that are used to basically ensure that the quality is of appropriate, is appropriate for the use of the data. Now, if you don't do QA/QC, and especially the specific tests, your data's garbage. Most people do know that, but they don't really think about it. And I mean, you really can't tell the difference between whether data is made up or whether it is authentically measured. So next slide. So when I'm explaining these concepts to my students, I tell them we have the QC trinity. We have the trinity consists of these three tests: a blank, replicates, and control. At a minimum, you want to have one of each of these per batch, where a batch is generally between 10 and 20 samples and no more than 20 samples in most cases. So if you could click 3 times. Thank you. So for blanks in the US EPA method, we have a reagent blank and a method blank. For the replicates, it's 1 sample analyzed in duplicate per batch. And then we have 3 control samples. The control samples are the ones where we have the known amount of analyte. And so we can basically judge how correct our measurement is. So we have the laboratory control standard, which is essentially a spiked blank that's taken through the entire procedure. We have a matrix spike, which is a known amount that's added to a sample itself that has been taken, the extract that's been taken through the procedure, and it's added right before the measurement, the analytical measurement. And then we have a standard reference material. And this is a soil material. So if you could click 3 times, maybe 4 times. Yeah. So these are, and one more time. So these are in red, the acceptance limits that are set out in the standard operating procedure of the method. If these acceptance limits are not met, it is instructed that you should repeat the analysis if possible, or qualify the data. So what I want to talk about is the standard reference material. So if you go to the next slide. The standard reference materials for bioaccessibility, for people who are not, directly in the measurement field, it's probably not that big of a deal that they exist. But in fact, it is, that we actually have acceptance limits that have been established for the specific bioaccessibility test for materials that are available to us and we can buy. So this was sort of a huge development when it came about. So what we use, or what is recommended in the method, is 2 standard reference materials from the National Institute of Standards and Technology, 2710A and 2711A. So 2710A is from essentially the same location as Montana High, if you're familiar with standard reference materials, was collected. And 2711A is from an agricultural field, also in Montana. Now, the acceptance limits that are established for these are for arsenic and lead for 2710A, but only for lead for 2711A. An interesting thing to notice, and maybe keep in the back of your mind as I continue through this talk, is that when the 2710A was collected, they found that the lead concentrations weren't quite high enough for what they wanted. So they added lead oxide to it. And this was to increase the lead concentration to where they wanted it. So the 2710A is essentially a material that is not completely authentically contaminated. It has had an amendment made. Next slide. Next slide. So the acceptance limits for NIST 2710A are shown here as control charts, and they are expressed in percent bioaccessibility. So this is the ratio of the bioaccessible concentration that's been extracted in that method that I talked about, divided by the total concentration and expressed as a percentage. So we have for lead, it's between 61 and 74%. It's pretty high. And for arsenic, between 33 and 49%. These are acceptance limits that have been established, that it's given in the SOP to use. And the lead one was based on a round robin experiment and 30 replicates. And the arsenic one was actually from 2 labs, but where they did 131 replicates between them. So next slide. And these are the results of what we have been getting since 2018. So for lead, we see the red dots, and for arsenic, we see the yellow dots. And what I'm sure is immediately obvious to you is that we are getting results that are, for the most part, lower than the acceptance limits. But what I would like to point out is that the blue dots that you see labeled lead 2011, these are the results that we got in our lab in 2011 for the round robin experiment. So when I mentioned that round robin experiment, we actually participated in that. And you can see that we actually got the numbers right where the acceptance limits are established. So at one time, we were getting the numbers that were within the acceptance limits. Now for arsenic, I don't have a comparison point.
3 minutes, Iris.
Sorry?
3 minutes.
3 minutes, okay, yep. So I don't really have a comparison point. So I just have a few literature values shown there. Next slide. So you might be saying, well, you're probably just doing it wrong. Well, that was what we thought too. But we did do a lot of repetition to find out what we were doing wrong. But we have been running 2711A samples as well. They've been coming right where they're supposed to be for lead. 27, we don't have a similar, we don't have the limits for arsenic. But what we do have is the 2710, which we've been running for the last 15 years in our lab. So we know where that's supposed to be for this method. And in fact, the numbers that we've been getting running these samples at the same time as the NIST 2710A are even a bit on the high side. So there's nothing to suggest that we are systematically running low with our method. And all other controls, blanks, and duplicates are within the limits. Next slide. So what's going on? Well, we don't really know. We've been hypothesizing that it might have something to do with the speciation. And so we're just sort of looking at that from a preliminary point of view. There's some speciation that was reported for the materials in 2011. You can see it's pretty similar between 2710A and 2710. The scorodite, ralg are a little bit higher, but those are pretty much insoluble species. So we don't really know what's going on and why it would change over time. The bottom profiles that you see there are from when Alistair looked at this continuous online leaching method. He looked at these materials. So this is a method that allows you to look at how the lead leaches out over time using the same extraction fluid. And we can see that there are profiles that are a little bit different for 2710A and 2710. But again, we don't really know what this means. Next slide. So to summarize, I hope that I showed you that health concerns are kind of where it starts for leading to the need for bioaccessibility measurements. And those bioaccessibility measurements need to have QC tests to enable them to be used in a risk assessment. And what we've been seeing is that our NIST 2710A results have been consistently low for both lead and arsenic. And we're wondering if it has to do with the stability of the material. I have shown the funding agencies, NSERC, National Defense, and Queens. And then my last slide is just a kind of a silly picture, maybe a good picture, I don't know.
Okay, thanks so much, Iris. So we'll go ahead and continue on into the lightning talks, and then we'll have some time for questions and answers for all of our speakers. So next up, we have Jessica Allingham, who's a PhD student looking at with a talk entitled Brightening Up Brain Injuries, the Design, Synthesis, and Characterization of PET diagnostic agent for neuronal trauma. So go ahead, Jessica.
Awesome. Can you guys hear me well? Good, perfect. So as Clay had just said, my name's Jess. I'm a PhD student at Lakehead University, and I want to brighten up brain injuries. So next slide, please. I'm from Canada. So the concussion statistics for Canada is that for every 3 minutes, 1 Canadian suffers from a brain injury. That adds up to 170,000 brain injuries every single year, resulting in 11,000 deaths and 6,000 individuals left permanently disabled. Next slide, please. Despite these facts, there is still no single standard objective criterion for diagnosing concussions. There is a glaring void in concussion diagnostics. Next slide, please. My research looks to fill this void by developing a fluorine-18 radio tracer capable of binding to a biomarker of neuronal trauma. Once it's bound, the buildup of that radio tracer can be imaged using a PET scanner, which will allow us to diagnose concussions by brightening them up. Next slide, please. So how am I going to go about doing this? So we're going to start with a severely injured brain. Click, please. As you can see right here, when you have an injury, astrocytes flock to the scene of the injury. Click, please. Just like first responders to the scene of an accident. I also like to think of these astrocytes like a scab. Click, please. Just like if you were to cut your arm and a scab would form to help the skin heal correctly, similarly, when you injure your brain, these astrocytes form a scaffold around which the cells and tissues can heal correctly. The only difference between the two is that a doctor can see the scab on your arm and know that you're injured, but they cannot see the scab in your brain. I want to make this brain scab visible. To do this, we looked at proteins associated with these astrocytes. Click, please. One of which is S100B. S100B is a protein associated with astrocytes that sheath blood vessels, and it's involved in inflammatory processes. When an individual receives an injury, click, please. The concentration and expression of S100B is significantly increased in the area of the injury. And it's that increase in protein concentration that I would like to image. Next slide, please. To target S100B, I employed virtual ligand screening to help come up with a structure that was most likely to bind to our protein of interest. Using this technique, we developed the suggested structure depicted at the bottom. And that was due to the fact that it looks similar to a known compound or a known ligand of S100B depicted at the top. Next slide, please. So the suggested structure was then synthesized using the outline synthetic scheme on this slide, wherein we combine 2 commercially available compounds, 1 and 2 on the screen, in a simple SN2 mechanism to produce our nitrointermediate, compound number 3 on the screen, which we then hydrogenated to form our primary amine precursor, which is compound number 4 on the screen. We then combined our precursor with a prosthetic group, fluoroethyl tosylate, and that's number 7 on the screen. This is a commonly used prosthetic group in radiolabeling because it lends itself well to N-alkylation, which is how we're going to be using it here. So in the final step, scheme 1C at the bottom, we combine our fluoroethyl tosylate prosthetic group with our primary amine precursor to give us our desired compound. And we were actually able to get that with a 62% yield. And so next slide, please. So we do have to make some adjustments when we move into radiosynthesis. We, instead of starting with that fluoroethanol that we did to produce our non-radioactive prosthetic group, we had to use bis-tosylate because we can't start with a fluorinated compound. So we're reacting a bis-tosylate, compound 11 here, with the fluoride ion, the fluoride 18 ion, to give us our radioactive 7 compound or fluoroethyl tosylate, so 7 star to indicate that it's radioactive. And then the second part of the reaction is exactly the same, where we're combining that prosthetic group with our primary amine precursor. And we were actually able to get a 58% radiochemical yield of compound 10 star, our actual radio tracer. Next slide, please.
30 seconds.
Thank you. So here we have just a radiochromatogram that proves that we synthesized the correct compound. And then the next slide, we have that we performed ELISA experiments, enzyme-linked immunosorbent assay, and we performed multiplets of this experiment and averaged the results, giving us a EC50 value of about 10 to 47 nanomolars. That's the 95% confidence interval. And this is great because it compares well to a known ligand of S100B. Next slide, please. And so for the future, we're going to move into cellular assays using astrocyte cells, which we know produce the S100B. And next slide. So with the successful completion of my research, we'll be able to diagnose concussions by brightening them up, as you can see in the image at the bottom, where we have a PET scan with a big bright red spot. And then finally, just my acknowledgements of my supervisors, my lab mates, the Thunder Bay Regional Health Research Institute, and Lakehead University, of course. And the final slide is just for questions. If you have any, there is my email address, and we can obviously chat at the end as well.
Great, thanks, Jessica. That's really cool work. Thanks for showing up and presenting today. So we'll get into questions here in just a few minutes. So we have next up Suzanne Charlesworth from the UK talking about sustainable drainage in refugee camps. And I don't know how to pronounce that word. You'll have to jump in there. Addressing human and environmental health. Thanks.
Okay. I've actually changed it, the title, so you don't have to say that word.
Okay.
It's changed to slums. Okay. Yes, so I'm going to be talking to you about the application of SUDS in refugee camps and slums. And this is in order to address human environmental health by tackling the lack of infrastructure for drainage and greywater management. The UN resolution says that WASH, or water sanitation and hygiene, is a human right, and that is access to clean, safe drinking water and sanitation. But what about drainage? Is that a human right? Drainage is hidden in sanitation, and it's generally called waste. Without drainage, you have flooding, proliferation of disease vectors, transmission of diseases like Zika, dengue, chikungunya, leading to negative impacts on human health, quality of life, and environmental degradation. So I want to talk to you about 3 case studies. Next slide, please. So this is the first one. This is from South Africa, and this focuses on the bioremediation of polluted river water, which flows through an informal settlement. The river itself is diverted through the bioremediation tanks, which you can see in the picture top right. One half of the tanks are vegetated, the other half is not. And the effluent that comes out of the bottom is diverted to irrigate small urban gardens like the picture bottom right. And preliminary results of analysis of vegetation and the soil indicates, and of the water, indicates more or less complete removal of E. coli. And if you compare that against the WHO treated wastewater for ready-to-eat produce guidelines, it has to be less than 1,000 coliform forming units per 100 mil, and it's way below that. Reduction of orthophosphates, conversion of ammonia to nitrates and nitrites, and in an analysis of 2 harvests from one of these small gardens of leafy and bulbous plants, it was found that they conformed to South African standards, and so were safe to eat. Also, the soils showed no signs of accumulation of either heavy metals or nutrient accumulation. Next slide, please. The second case study is of a refugee camp from the Kurdistan region of Iraq. And the middle picture shows you a resident of a camp harvesting grey water, which has been disposed of in an open channel, and they're going to use that to irrigate plants around their shelter. Now, if you take a look at the table above that photograph at the top, this shows the initial water quality for a site that we got from UNHCR. And this is the analysis before construction of a sustainable drainage management train. So there are two inlets, sample 1 and sample 2, and an outlet. And if you concentrate on, again, on the fecal coliforms, and again on the WHO treated wastewater guidelines, you can see that actually, that again, these are very, very high. The SUDS management train, which is at the bottom left of the picture, was designed, constructed, and is now managed by the refugee community. Unfortunately, due to COVID, we've been unable to go back and re-monitor that site and see if the water quality has improved. But the community have taken over that site. So can you press once, please? Yeah, they've planted edible crops at the site, and they've also, those little buildings are chicken sheds, sheep, places for sheep to go, goats,
30
and
seconds,
geese.
Sue.
Oh lordy, next slide please. So then the final case study is to stop the mosquito that carries the Zika virus from breeding. So the left photograph is of a puddle outside of a favela dwelling. It hadn't rained, that puddle was full of mosquitoes. On the right hand side, you can see the problem with solid waste and with water accumulating amongst it. You can do something simple with that. Next, press once please, by installing a gravel-filled trench with potentially a grass filter strip, and that would stop the mosquito having access to the surface of the water. Next slide, please. So technically we can do it. You need to have community engagement or it will fail, although in a favela that's quite difficult. Kevin Winter from the University of Cape Town says this is socially responsive drainage for which you have accountability. You encourage the community to participate, in which they are included, and whatever you do is transparent. Press once, please. So can you go from wash to washed, include drainage? Press again, please. And is drainage a human right? Sorry, I've gone over.
Thanks so much, Sue. That was really interesting. Thanks for those case studies. It seems like you've been doing some really good field work. Okay, so next up we have Annalise Gorino, who is going to be giving a talk entitled Estimating Radon Fluxes and Different Data Sets. Different data sets to discriminate sources and assess human health risks in Campania region of Italy. So go ahead.
Thank you. Good afternoon. I'm glad to be here with you today. Next slide, please. Radon is an ubiquitous radioactive gas proceeding from the decay of some naturally occurring radionuclides. It can accumulate indoor and represent the main source of natural radiation to which human beings can be exposed. Radon itself is chemically inert, but its progeny is very reactive and decays, emitting alpha particles that, if inhaled, can damage DNA. In fact, radon is considered the second indirect leading cause of lung cancer. Click one, please. The objectives of the study were to estimate and compare radon fluxes resulting from two different data sets, analyze and interpret the differences, and identify areas where with a high exposure to radon for humans. Next slide, please. Sorry. Campania is a region of the southwestern sector of the Italian peninsula, whose territory is mainly characterized by the presence of volcanic lithotypes and sediments, in which the radionuclides content is generally high. Next, please. Using radiometric and geochemical data recorded in two different prospective campaigns, some empirical relationship, please click 3 times, were applied to estimate the radon flux across the region. This also involved the calculation of the terrestrial gamma dose rate, which represents the total gamma radiation emitted by the decay of naturally occurring radionuclides. The color composite maps here show the spatial distribution of radionuclides association. I'm sorry. High values represented by the brightest color correspond well with the volcanic centers in the central western sector of the region. Next, please. All data were processed in a GIS environment and were interpolated by means of the multifractal inverse distance weighted algorithm, and the grids were classified to the concentration area fractal model. Radon flux values reached, as expected, their maximum in correspondence with the volcanic centers, whereas low values occur in the inner sector. Click, please. Considering a threshold limit for prevention and control of 50 millibecquerel per square meter second, it was possible to identify an area of about 90 square kilometers on the southwestern sector of the Monte Somma Vesuvio, which correspond to an area characterized by a very high population density, and in addition is known to be featured by high lung cancer incidence. Next, please. The resulting flux values were compared, performing a regression analysis to assess the functional relationship among them, and residuals were used to generate a map of their distribution. Further, by means of a software that allowed to compare the map pixel by pixel, a similarity map was produced. The analysis of residuals and similarities suggested the hypothesis in the volcanic area where the residuals are markedly positive, so that's red, and the maps show a quite good similarity, the pink values, of an underground contribution to the potential superficial radon flux due to the presence of deep volcanic thick layers, whereas in the eastern mountainous area, where the residuals are markedly negative, except for two small regions, and the maps result totally distinct. So in the map to the right, the red color, these differences were attributable to the presence of tectonic structure. Next, please. So in conclusion, we can assess that the study led to discriminate the nature of the different sources that potentially contribute to the superficial radon fluxes, and a great contribution to the high values was attributed to some regional areas characterized by the presence of deep volcanic layers and active tectonic structures. And if the proposed techniques will be verified by field measurement, this can make it possible also in area where direct measurement of radon fluxes will not be possible, to have a tool for managing the areas in which the health risk for the local population could be relevant, and for the individuation of hidden geological feature. Next, please. So thank you for your time, and I will be glad to answer your question, if any, and click. For further details, I invite you to consult our recently published work on chemosphere. Thank you.
All right. Thank you so much, Annalise. That was a great talk, right on time. Perfect. Let's start with questions. Does anybody have a question for Annalise? So you can either ask it in the chat or just go ahead and jump in. Just make sure and unmute yourself. Anybody? So what is like these concentrations of radon, are these, I'm just trying to get an idea of, they're really high, are they super toxic and dangerous to humans and wildlife, or are they sort of trace amounts? Just, yeah, what's the kind of toxicity level of these measurements?
For Italy, there are no regulation on the amount of radon, neither for the flux, neither for the concentration in becquerel per cube meters.
Okay.
The limit we used of 50 millibecquerel per square meter second is came from a Chinese regulation, because we found a Russian regulation and a Chinese one, but the Chinese one was the most conservative. The Russian was about 80, so we used the more conservative one to assess the risk. But in Italy nowadays there are no regulations.
Great. Yeah. And you said something about ground measurements. Do you all have plans to follow this up with any additional measurements on the ground, or that's for someone else to do in the future?
We started to do measurement in the Campi Flegrei Flegrei fields volcanic area. We started measure the content of radon in water.
Okay.
And then for the next month, we plan to measure the radon flux in soils.
Great. Well, thank you so much. That was great. So does
to
anyone
you.
have a question for any of our other speakers? So feel free to just kind of, like I said, throw one in the chat or just jump in. We had some really good talks.
I was monitoring the chat throughout the
Yep.
presentations, and there haven't been any questions yet. But if you do have them, you can put them in or just raise your hand.
Okay. I was interested, David, are you still on the call?
On the call, yep.
Yeah, thanks for your talk. I really enjoyed it. I was just, sort of wondering, and maybe you said this and I didn't get it. Does the chlorination affect the toxicity in the environment? Because you said something about the what ionic silver was the worst. Does chlorine binding to it sort of detoxify it at all?
Yeah, so it's a fantastic question. So silver chloride does indeed reduce the toxicity because it reduces its solubility in water.
Okay.
And in theory, it makes it harder for it to be released in subsequent events. However, people have started putting silver chloride into textiles for the new generation.
I'd heard of that, yeah.
Yeah, and those still do inhibit bacteria. And in the presence of water and sunlight, silver chloride can convert back into metallic silver.
Okay.
To answer your question shortly, yes, it does reduce toxicity. However, it's a little more complicated than that.
Right. So maybe like even like the sweat binding it up, once it gets out into the water column or something, like in a river or lake or something, then like the light could sort of disrupt that and turn it back. So there's a lot of processing that happens. That's really interesting from, thinking about, managing for health or something like that, because it's sort of a constant, the way I'm seeing it in my head, it's kind of like a constant turnover between it's bound to something, if it's charged, it might bind to another particle and desorb. So once it's there, I guess there's a lot of complex chemistry that could have health implications.
Yeah, absolutely. We fortunately, as part of the group, we looked at the impact of the silver materials on soil and wetland systems.
Yes.
And of all this silver that we studied, the only one that had impact were high concentrations of ionic silver.
Yeah.
The main reasons for that is oxygen, sulfur, chlorine as well. All these things love to just grab onto silver and knock it out.
Yeah.
As soon as you start to get to wetland systems where you have biofilm just loves to remove silver and just kind of sequester it off to the side. So
yeah,
this is one of those rare situations where it might be a good news story in the sense that, like, maybe nothing bad happens, but I think more study is definitely required.
Great. And thanks for that answer. That was really great. Does anyone have any questions for our other speakers?
I actually had a question for Professor Charlesworth. You were talking about how these gardens grew vegetables that had no contamination and they were perfectly safe to eat according to the standards. I'm wondering if the gardens that were the filters in that kind of first section that you talked about, is it the soils and the beds that the wastewater is flowing through that's actually kind of cleaning the wastewater, or is it actually the plants that are planted in there that are maybe cleaning it as well? I'm just curious what's contributing to that.
It's two things. It's both things. It's the substrate and it's the plants. But actually, the previous David, was it his name, mentioned biofilm. And there's a biofilm that develops on the aggregate, and it also develops around the roots of the plants. And similar to what he was talking about with the silver, the biofilm either metabolizes nutrients or it will trap particulate associated pollutants. So it's actually a combination. The problem is with wetland plants, if you've got seasonality and then they die off and they've taken stuff up, then that can be a bit of an issue. It might need some extra maintenance and possibly in the winter when they've died off, they're less efficient. But if you've got a good biofilm developed on an aggregate, it can really take up a lot of pollutants. And we found that in pervious pavement systems. A long answer to a short question.
It's really interesting. No, that is really interesting. Thank you.
Thanks, Sue. Any other questions? Does anyone have anything for Jessica? The chemistry was definitely a bit over my head, but I really enjoyed it. And I think it's kind of a nice, sort of different use of some of these techniques, right? So we're trying to, measure how these things change through the environment. She's actually targeting stuff to attach to a protein to sort of light up for human health complications. I just didn't know if there's maybe any sort of overlap between anyone else, interest or expertise in herbs, because I thought it was like a great, applied sort of technique for doing this, and it was really, really exciting because obviously brain injuries are really bad. So does anybody else have anything for her before we go into a quick coffee break? Or any other general questions? I might have to email Jessica later. I have more questions. Okay, so why don't we go ahead and take a quick 5-minute break? So we ran over into coffee period, but that was good. It was sort of designed that way. So yeah, so the first session, I sort of set it up, to be more, I guess, super in line with, SEGH Geochemistry and Health. But I also had to, bother a lot of other people, sort of my friends who do a bit more ecological health, to give some talks in this. So I kind of set it up. So yeah, we'll sort of switch maybe a bit more from human health to more ecological stuff in the next session. So please stick around, come back. I'm going to mute myself, but feel free to keep chatting. I don't know if Michael or Bob have anything to say.
Not really. Much of this is beyond me. The simple geochemist. But I think things are going really well. Some very interesting, informative, and important presentations.
Yeah, I feel like I owe Malcolm some tuition fees or something, because he gave me a really good background in a crash course and how all of this stuff is done. So I really appreciate that. That was great.
Just to add, thanks very much to the speakers. We've got a nice diverse range of talks, which is very typical of SEGH. So yeah, wonderful.
Yeah, you always learn something when you tune into one of these. So hopefully we can keep them going on. All right, so we'll go a few minutes and then we'll start back on time. Okay, well, that was a short break, but Alistair, are you there? Do you want to pull up the next talk and
Yeah, I can get it queued
see if
up.
we can get, we'll see if we can get Sally.
Well, I'm here. Can you hear and see me?
Yeah, you're great. So I'm really excited to, we started off the first session with sort of a really good overview of medical geology, and Sally has a lot of work in this field. So she's done a lot of work before I met her when she was doing some shale work and gas drilling in Arkansas. And now she's since moved to Virginia Tech, and she's getting into salt. So I'm really excited to see Sally's talk about the effects of salt on carbon quality and cycling across riparian stream boundaries. So take it away, Sally.
Thanks, Clay. I really appreciate the invitation, and I'm learning a lot. I think that we should consider more in our own research. I want to point out that this is a collaborative project, so I'm presenting many people's data. Next slide, please. And that includes Natalie Clay at Louisiana Tech and Michelle Evans White at University of Arkansas, and Connor Gruntz and Maggie Herman. They were graduate students. So I just want to point out that this is not just my own work. Next slide. We all know that we're salting the earth. We're bringing salt up from the ground and putting it everywhere. Road salting, irrigation, wastewater, resource extraction that exposes rocks. So then they dissolve, and we get major ions that are in the soils and in fresh water. And this poses some complications for the organisms that live there. Next slide. And one of the ways that major ions, or I'll just say specific conductivity, has been shown to increase in fresh waters is through gas extraction using hydraulic fracturing. And this could happen in two ways. One would be just the land clearing itself will expose more rock and sediment to weathering, and you get more major ions. But the other way is just deep hole drilling exposes more rock and even brings up brines that are held on the surface of close to streams, and that can sometimes enter the adjacent streams. So next slide. One thing that we found in our research working in central Arkansas, so pretty much in the middle of the United States, lower 48, if you look at those red dots, those are oil and gas wells that were spud from about 2005 to 2010. And what we did was we measured the natural gas well density shown on the x-axis and these major elements that are shown on the y and conductivity. And what you see is that there's a really strong positive relationship. But you'll also note that these aren't very high concentrations to begin with. Next slide. But this trend of seeing increases in major elements and specific conductivity as kind of a bulk estimate of increased ions in fresh water is a global trend, and Sujay Kaushал's work and others have shown that it's happening all over the lower 48 states, and there are multiple different sources of these ions. Oftentimes it's a combination of urbanization, infrastructure, road de-icing, mining, so resource extraction. Next slide. And from a freshwater ecologist perspective, we think about how this is going to kill aquatic organisms and cause impaired aquatic life. And in the United States, the Clean Water Act recognizes that as a violation, and there can be regulatory actions from that. But we were really interested more in how it changes the ecology and the processing of the streams themselves. So what we knew about Arkansas was that at a electrical conductance or conductivity of about 328 microsiemens per centimeter, we lose about 10% or 5%, excuse me, of our macroinvertebrate genera. And of those genera, some of them are really important in carbon cycling. And so what we wanted to know was if these small increases in conductivity was affecting carbon cycling by changing the organisms or their performance in these fresh waters. Next slide. And I was just thinking about it as, yeah, it's bad. We don't want to see the rise in these ions because it's going to lead to loss of species or at least a reduced performance. But Natalie Clay was listening to one of our students' talks, and she's like, well, those are really low concentrations. I've been working in the tropics in the terrestrial ecosystem, and we know that adding urine, which is full of major ions, will increase decomposition rates, and you can get an increase in arthropod detritivores. So it's nutritive. Next slide. So from the riparian perspective, especially in low salt deposition areas, they consider adding salt as a nutrient because it satisfies consumer demand like plants cannot. So you get an increase in decomposition rates. In aquatic systems, we consider it bad because of the constraints that aquatic organisms have on regulating their ions. They're very salty compared to the outside water. And if the external environment becomes more salty, it's going to increase their energy allocation towards regulating ions and water balance. Next slide. And so we put our brains together, the three of us, and we came up with this conceptualization of how we think that salt entering a riparian area could affect streams and vice versa. So salt just enters, let's say they dump it on a road. It flows into a stream and it's going to affect directly the organisms that live there that process carbon. And we predicted it would be detrimental. But if we look at pathway 2, we know plants, while they don't need these ions, they store them in their vacuoles. They become salty themselves, fall into a stream or into the riparian area. And that could either satisfy a nutrient demand or it could lead to stress. And we weren't sure which one. But the third pathway is the salt just falls onto the, it just like in irrigation, it just accumulates from evapotranspiration and it affects the microbes and detritivores probably first by being nutritive up to some point before it becomes toxic. Next slide. And so Natalie did a salt addition to riparian area in northern Louisiana. It's the bottom close to that red area, the Gulf. So there is some salt already in the soils. Next slide. But what she did was end up looking at pathway 2, where they predicted that when they salted when they added experimentally the salt to the riparian area, they would have salty leaves that would increase detrital processing, or just direct application would stimulate microbial and arthropod decomposition to also stimulate detrital processing. So positive, positive. Next slide, please.
2 minutes.
And what they found is that why don't we just go to the next slide and I'll tell you what they found. Okay, so what they found was that the decomposition rates were lower in the salt added plots compared to the just watered plots. So this was contrary to their prediction. So the microbes were evidently not salt limited. Next slide. And they did find that they were able to increase the amount of sodium, the salt, in the riparian plants. So they did actively take it up. And so that suggests that we will have saltier detritus. And so now I'm just going to jump a few slides. Next slide, next slide, next slide, next slide, next slide, and next slide, next slide, next slide. Okay, so I messed up the time. I thought I had 15 minutes and I evidently don't because I'm at 8:26 here. So in the riparian area, what happened was they did see that the microbes declined, but the plants took up salt. So there wasn't sodium limitation. And this may be because of where they were measuring the salts. there was already enough salt in the soils. Now in streams, we took a slightly different approach because these results didn't scale up to the stream level. So next slide.
Sally, I'm so sorry. I did mess up there. I don't know if we could go back. I can give you my time. I really apologize for that.
Nope, that's okay. And so now we took it to the microcosm level, and what we did was we manipulated the salt concentrations in microcosms. And what we wanted to know is if the shredders, the aquatic shredders, would show that same sodium limitation and increase their growth. So they changing their performance and increasing the ecosystem functioning measured as decomposition, or if it would stress them out because of that ion water balance stress so that it decreased decomposition. So while with the field experiment, we didn't see anything happening in the stream, probably because of the issue of scale. We were just adding salt with a backpack sprayer, not enough to see a stream effect. So now we're going little, next slide. And we're going little, and we're predicting that if we add salts to stream water and across a gradient, that we're going to see an increase in our aquatic invertebrate, we call them shredders, they're eating leaves, in their growth up to some type of threshold before they become nutrient limited, because first they're nutrient limited, then they're osmotically stressed. And we predict that the same for decomposition, because the macroinvertebrate shredders are really mediating or governing that process. Next slide. And so we had a gradient of sodium chloride that ranged from 16 to 64, and we had ambient sodium chloride concentrations in our stream water. And there's our little bug and our little pea cups, and we just exposed them to these salts directly, and we fed them leaves and looked at decomposition rates. Next slide. And so what we found, I'm showing this as an effect size. So if a value is above 0, it means there was a subsidy effect. And if it's below 0, it was stress. And if it's at 0, there was no effect. And what we see is that the Amphinemura end up showing a subsidy effect if growth is the response variable here. So, and there was no stress. So we didn't achieve the hump. We just achieved the left hump side of the figure. Next slide. And then similarly, we saw, and it looked like at the decomposition tended to increase, but it wasn't that strong of an effect. So next slide. So decomposition isn't just driven by the macroinvertebrate shredders, it's also driven by fungi, and the fungal biomass actually really increased too. So it looked like we had a subsidy response by both the macroinvertebrates and by the heterotrophic microbes. Next slide. And so we've done these types of experiments at small scales for many different macroinvertebrates. Sometimes we see subsidy, sometimes we see stress, and we don't know what's physiologically really mitigating their differential responses, because these are across the same salt gradient. Next slide. But one thing that's also cool is we're seeing that the bug itself, the insect, is accumulating salt. And in this case, when I say salt, it's sodium, and that has consequences for reciprocal flows. Next slide. And then finally, we're trying to put this all together into a more comprehensive framework, where what we think is going to happen is if salt limitation is driving carbon cycling in the system, we can have more efficient carbon cycling and tighter coupling between the riparian stream area. But if toxicity responses prevail, it'll decouple that riparian stream interaction. And so that's what we're working on right now. And I'm sorry for the mix up in the time. I'm done.
No, that was totally on me. And we still have a bit of time for questions. And I apologize. I thought we'd started 5 minutes earlier. I didn't go by my own schedule. So
that's okay. No problem.
That's really awesome work. And I wasn't aware of most of this. So I really love seeing it. Thanks so much. Does anybody have a question for Sally? So I'm super interested in salts as well, but not necessarily from like Well, no, both concentration and composition, right? Because we have all these different types of salts, you know, like you said, coming from all these different types of areas. Is there any work looking, I guess, at sort of toxicity versus nutrition effects across these different types of geographic areas or you know, applications wherever it's coming from, because like there's, you know, tons of different cations that are attached to those chlorides, and they have different toxicity effects and nutritive values as well. I think there's so much work that could be done.
There is. It's endless. I think that Chris Neach at the EPA ORD has done a lot of cocktail experiments in large scale flume. So his work is one person's to look at. And then Mount, he's works for the EPA, has also done a lot of mixtures and trying to parse out the cation anion effects. And there's a new paper, I think it came out in 2018, that it's like 50 studies to really try to understand which is more, you know, what is it, the anion that's more detrimental? And if so, you know, what's the effect size of that? So our work really is, we're working at really low concentrations. And I think we're just trying to start at this riparian stream interface, and sodium made sense to us to start with sodium chloride because it's pervasive, and we know sodium is strongly regulating decomposition in like tropical regions.
Yeah, I like Natalie's work and like everything coming out of OU, so that's really cool. Okay, so yeah, if you don't hate me for messing up your time flow, I'll have to talk to you a bit more later.
Sounds good.
So next up, we have Nicole Wagner from Baylor University, and she's going to be talking about acclimation to variable nitrogen to phosphorus ratios and how it affects the ionomic composition and physiology of nitrogen fixing cyanobacteria. Nicole?
Thanks, Clay, for the intro.
Starting my timer right now so I don't mess you up.
Okay. I'm fine if you cut me off. I'll pay you back some other time. I'll cut you off during your talk.
Okay.
I guess, yeah, so next slide. This is this project actually is in collaboration with Clay and his advisor, Puni at OSU. So to get sort of at the human health aspect, harmful algal blooms are really common, and they're potentially increasing in frequency and duration caused by nutrient pollution and climate change. And so this graph here is from a paper from 2016 looking at the global distribution of microcystis, which is a really common harmful algal bloom that occurs globally. So everywhere on this globe, on this figure, you can see the green, that is where microcystis has been identified, and also there has been toxins present. And then in the blue areas, which there isn't many, that is just that microcystis has been identified, but the blooms may have not been toxic, or just toxins weren't measured. And so we know these are occurring all over the world, but we actually really don't know how they're formed, and that's really poorly understood. Next slide. And so what we do know about harmful algal blooms in general is they are correlated really well with high concentrations of total nitrogen and total phosphorus. So this is a paper from Yuan and Pollard from the EPA, where they took the 2012 National Lakes Assessment data, where they go out and they sample a bunch of lakes over a really short period of time in the summer. And they looked at the concentrations of total nitrogen, which is on the x-axis, and total phosphorus on the y-axis. And they looked at microcystin again, so this toxin that the cyanobacteria can produce. And what they found was basically under 100 micrograms per liter of total nitrogen, you more or less aren't finding this toxin, but over 1,000 micrograms per liter of total nitrogen, this cyanotoxin is really prevalent. And also, as you increase the total phosphorus concentration, it also becomes really prevalent. So we know that basically cyanobacteria, not just toxins, but their overall biomass, seem to increase as we increase total nitrogen and total phosphorus, or the conditions become more eutrophic. Next slide. And so what else can this data sort of show us? So looking, this is basically the same figure, except for instead of looking at microcystin, the toxin, this is just looking at total nitrogen and total phosphorus, and then sort of getting at how these elements change. And so using this Black line, this is an NDP, so this is the amount of nitrogen per phosphorus. That is an NDP of 23 by mass, or this is by 50 by moles. And anything below that line is could be classified as phosphorus limited. And then the red line is NDP of 9 or NDP of 20 by moles. And anything above that line would be nitrogen limited. And then in between, we actually have what's classified as balanced or co-limited growth. And so when you add up all the lakes from the NLA database, it was found that around 14.2% could be classified as nitrogen limited and 17% could be classified as phosphorus limited. So nutritional imbalances are fairly common within lakes. This is just within America, but also globally. Next slide. And so how do cyanobacteria deal with imbalances? So there's two really main mechanisms, is that they can increase their nutrient uptake. And can you hit the space bar or however you proceed? Yeah. And so here you have this is just a schematic, and the nutrient uptake transporters, they can increase of the limiting nutrients. So then you have more spaces where you can capture more of this limiting nutrient. Next. And the other way that you can sort of deal with imbalances is you can actually substitute the limiting nutrient for non-limiting nutrients. And so cyanobacteria, especially in the marine environment, they can actually substitute out their phosphorus in the phospholipids for either galacto or sulfolipids. Another common example is within enzymes that have a metallic center. Coenzymes, you can potentially sub that out. Example, iron could sometimes be substituted out for copper and zinc in certain enzymes. And the other way that cyanobacteria can deal with nutrient imbalances, especially dealing with nitrogen, next slide, is some cyanobacteria are able to actually fix atmospheric nitrogen. So they can break that triple bond into two usable ammonia molecules, which then can be incorporated into proteins and used for growth. So these cyanobacteria will use the combined nitrogen sources first. So this is like nitrate, nitrite, ammonia, or urea, or other dissolved organic nitrogen compounds. But after these sources are exhausted, the cell recognizes it and it creates a heterocyst, which is the circular cell in the middle. And this is where the site of N fixation happens. So nitrogen fixation occurs through the use of a nitrogenase enzyme. And this is actually a really oxygen sensitive enzyme. And so this cell basically prevents oxygen from crossing the membrane to keeping it active. And then once these cells are formed on the filament, they basically remain there, especially within this species. And then if nitrogen is reestablished by like nitrate or urea, whatever, then the nitrogen fixation stops. And next slide. And the other thing that sort of can affect this nitrogen fixation is also looking at the environmental history. So we did a previous study with a PhD student in our lab, Felicia Osborne. And what we have found was that looking at the two red boxes on day 21 and day 28. So we grew these cultures of this N-fixing cyanobacteria under different nitrogen imbalances, which is the x-axis. So the negative numbers means that nitrogen is limiting, and the positive numbers on the x-axis means that nitrogen is in excess. And then on the y-axis, we have the amount of nitrogen that was fixed just in micrograms during time. And we acclimated these nitrogen fixing cyanobacteria to low nitrogen in the gray and high nitrogen in the black bars or black circles. And really what we found is after 21 and 28 days, the ones that were acclimated to low nitrogen conditions, so would be prime for N fixation, they actually were able to fix more nitrogen than the ones that were acclimated to high N conditions and not prime for N fixation. Next slide. So this really led to my question of looking at this acclimation a little bit more and how different NDPs not only affect just nitrogen and phosphorus, but also other ionome. So looking at the sodium and potassium and iron and magnesium, and how does that also affect physiology? Next slide. And so what we hypothesize is that the NDP growth conditions will alter their ionome and pigment concentrations. So this is like chlorophyll and phycobilins, and this is going to be regardless of the acclimation. So we know that the supply of nutrients should really affect their ionome. And then additionally, in low NDP conditions, the ones that were acclimated to low nitrogen to phosphorus, they'll be primed for N fixation, just as our previous studies showed. And so to be primed for nitrogen fixation, we expect them to have more nitrogen because they can fix more. We also expect them to have higher amounts of light harvesting pigments because we know nitrogen fixation is a really energetic process, and the metals associated with acquiring nitrogen. Next slide. So our acclimation period, this is the strain we used was the Dolichospermum flossaqua that we obtained from University of Texas, and we acclimated them to 4 different nitrogen to phosphorus conditions. So we had 2 that were extreme N limitation and moderately N limited. This is the N to P 2 and 10, and then phosphorus limited and extreme phosphorus limited. This is the 50 and 70, that should say 6. And we separated these in September of 2017, and we've been growing them in these cultures ever since. So at the time of this experiment was completed, it was around 1,000 generations were done before this experiment. Next slide. And so what we did is we took these acclimated cultures and we did almost a factorial design where we crossed them. So the N to Ps, the acclimated cultures were grown in N to P 2, 10, 20, and 50. So again, that ranged from nitrogen to phosphorus limited. And then we measured the ionome, the chlorophyll, and the phycobilins. And chlorophyll A and phycobilins are what they use for their light harvesting capabilities. Next slide. And so here's just the biomass results. And in the top panels, these are what they were growing in, and then each dot is the acclimation. So on the x-axis, we have acclimated in 2, 10, 50, and 76. And then on the y-axis, we have how much biomass as carbon they produced. And basically between acclimations, there was more or less no difference that we found. Within growth conditions, we did find differences. So the ones that were grown in N to P2, they grew less than all the other treatments. And that's because nitrogen fixation is really energetically costly and they can't grow as well. But within, between the acclimations, they didn't really grow that differently. Next slide. And then, so how did this affect their ionome? So this graph is a principal component analysis. So we took all of the ionomic data that we had and we compressed it into 2 dimensions. And so we have a principal component 1 and principal component 2. And so overall, we were able to explain about 60% of the variation. And then how you interpret this graph is based, the colors that you see, if they're grouped closer together, their ionomes are more alike. And if they're further apart, their ionomes are more different. And so we see this really clear separation in the red and the green. That's the ones that were grown in N to P 2 and 10. Compared to the N to P 20 and 50 that were clearly separated. So there's a good separation between the growth conditions. Next slide. And so this principal component analysis, it sort of gives you on how the ions are separated. And so the ones that were grown in low N conditions, they appeared to have more boron, magnesium, and calcium compared to the ones that were grown in high N conditions that had more nitrogen and sodium. Next slide. And so now we're just going to look at sort of how these acclimations potentially changed. So this is all grown in N to P2. So this is all under N limitation. And our data was standardized to carbon here, which I forgot to mention for the PCA as well. And basically how you interpret this is if the data appears higher, there's more of that nitrogen or molybdenum, whatever it is, in those cells compared to the ones that are lower. And so yes, on the x-axis, we have the acclimation. And so what we found here is that the ones that were acclimated to low N to P conditions, so prime for N fixation, tended to produce more nitrogen as what we hypothesized, more molybdenum, and actually had less calcium and less magnesium compared to the ones that were acclimated to the high N to P conditions. So acclimation really does affect their ionome. Next slide.
3 minutes, Nicole.
Okay. And then just looking at the exact same constant or the exact same ions here in ones that were grown in N to P 50 conditions. And so basically what we're finding is that under N limited conditions that we just saw, there was really big differences caused by acclimation. But when we grew them under N rich conditions, those acclimation differences disappeared. So there's really no difference in nitrogen or molybdenum, calcium, but the difference in magnesium remained. when the ones that were acclimated to low N-to-P conditions had less magnesium compared to the ones that were acclimated to high N-to-P conditions. Next slide. And then sort of to look at the pigments. So this is chlorophyll and phycobilins. So the top two panels are ones that were grown in N-to-P2, so nitrogen limited, and the bottom two panels are ones that were grown in N-to-P50, so nitrogen rich. And the ones on the left are the chlorophyll standardized by carbon, and the ones on the right are phycobilins standardized to carbon. And really under low conditions, just like the ionome acclimations, we've seen that there's more chlorophyll and more phycobilins compared to the ones that were acclimated in the high N conditions. And then under N-to-P50 growth conditions, these acclimation differences sort of disappear. Next slide. And so really, what does this all mean? So these changes in macronutrient supply, yes, obviously it alters both the nitrogen and phosphorus, but it also alters a lot of these other ions. And this could have drastic effects on micronutrient cycles that people aren't really paying attention to. And we don't really know about the frequency of micronutrient limitation because we tend not to measure it. And we don't know how this will affect harmful algal blooms, either growth or the potential of toxin production. And then just looking at the acclimations, we don't know how these acclimations will also alter the biogeochemical cycles. And I don't know what I was going with there with the last point there. But yeah, so the low phosphorus acclimated cells, they seem to have more magnesium regardless of growth conditions. And if anybody potentially knows about cyanobacteria physiology and could have a reason for that, I'd love to know why. But the low acclimated N conditions, we know they do have more pigments, molybdenum and calcium, and these are also used to help fix nitrogen. So that one was sort of obvious. Next slide. And yeah, with that, I would just like to acknowledge our funding sources through our NIH Oceans and Human Health Center and all our lab mates that were vital for helping this experiment.
All right, thanks, Nicole. That was right on time. We'll go for some questions a little bit later, but it's really fun to see those results. We can chat about magnesium at some point, maybe. Okay, so next up, we have our last 15-minute speaker. So we have Marie Aurora Armienta giving a talk on arsenic and chromium in groundwater of central Mexico, identification of sources and alternatives to avoid population exposure.
Okay. Well, good afternoon, and thank you very much, the organizers. I will talk about this presence of arsenic and chromium in Mexico. Next, please. Well, groundwater supplies more than 60% of drinking water of the country. Next. Some of the problems that have been identified in groundwaters are saline intrusion, nitrates, coliforms, and arsenic, fluoride, iron, manganese, and chromium, besides chromium, organic compounds. Next. Well, as you know, arsenic is one of the most toxic elements, and it causes many health issues. Next. And next. The toxicity varies among the compounds and oxidation state of arsenic, being arsenic 5 less toxic than arsenic 3. And there is a HACRE that the health produced by ingestion of arsenic in water or food. Next. Well, there are a list of some of the arsenic health effects like peripheral vascular disease and cytogenetic damage, diabetes, and cancer. Next. Well, the arsenic species in natural waters depend on the oxidation, the degree state, and the pH, the redox condition of the media. And as you see, at high pH and 6 to 8 pH, it exists arsenic 5 as an ion and arsenic 3 as a non-charged species. Next. This is important since anion arsenate as an ion may absorb on iron oxidroxides and other clays and calcites and other aquifer materials, and this changes its mobility in the aquifers. Okay, next. The first place where arsenic health effects was identified is La Comarca Lagunera, north central Mexico in 1958, with arsenic concentrations up to 0.7 milligrams per liter. Since then, next, many research has been performed. Well, volcanic and geothermal zones are other areas where arsenic may occur at high concentrations, and Mexico has many active volcanoes and also geothermal areas. Next. Well, in central Mexico, there is Los Humeros geothermal field, where up to 162 milligrams per liter of arsenic were determined in geothermal wells. Next. And mineralization is another potential source of arsenic to groundwater. And as you see in this map, Mexico has many mineralized zones with many metals, like lead, zinc, and silver. Next. And in this processing, extraction and processing of these ores have produced wealth along more than 400 years, but also has produced waste. Next. Well, there have been reported more than 240 arsenic-bearing minerals in the world. Next. And in Mexico, there are the occurrence is important, like arsenopyrite and mimetite and scordite in several places. Next. And this reflects the place that Mexico had in 2007, being the 9th place world arsenic producer of the world. Next. Well, as I said, the extraction and processing of minerals next produce the waste. Next. That are in many parts of the country, millions of tons of this waste, tailings. Next. And they may pollute the soils and produce solutions named acid mine drainage with high concentrations of arsenic and heavy metals. Next. Well, we developed a study at Simapan, an historical and current mining area since more than 20 years now. And next, this is in central Mexico, near Mexico City, about 250 kilometers. And as you see, the concentration of arsenic reached up to 1.2 milligrams per liter or more sometimes. And in one of the wells, in the most polluted one, but others also had very high concentrations. They mixed these waters with other wells, non-polluted. And finally, the red dots corresponds to drinking water that is supplied to the population. And in some occasions, it was very well above drinking water standard. This induced the consumption of these waters, induced the hyperkeratosis, hypopigmentation, diabetes, and other health problems. Next. Next, please. Well, okay, the geology corresponds mainly to Cretaceous limestones, and the mineralization is lead, zinc, and silver high temperature carbonate-hosted Mexican ore deposits. And gangue minerals include arsenopyrite and pyrite, among others minerals. Next. Well, this is a very semi-arid zone with no superficial water bodies available. Next. Okay, tailings are very close to the town, and when we started this study, many people, including us, thought that those were the source of arsenic. Next. Next. We developed environmental studies, including the geology, hydrogeology, mineralogy, and sampling and analysis of different types of materials like water, soil, etc. Next. We sampled tailings. Next. At depth. And analyzed in the tailings. Next. The chemistry and the mineralogy, and differentiate the total arsenic from the water soluble arsenic and that released by sequential extractions. And as you see, mineralogy is different, show difference between red tailings, oxidized and non-oxidized tailings. Next. Next. Well, the shallow wells near those tailings, yes, next, show some physicochemical characteristics, is particular, like an high sulfate concentration in relation with other anions that we expected that bicarbonates were more abundant, but they weren't, and arsenic related with sulfate very well. Next. This led us to think of the possible source of arsenic, and this is acid mine drainage that oxidizes in the tailings. Pyrite is oxidized, and also arsenopyrite releasing sulfate and also arsenic. Next. Next. Also a smelter that operated at Simapan until the mid of the last century released the arsenic-rich fumes. Next, that polluted the nearby soils with total arsenic and also with soluble arsenic that was present about almost 60 years after they terminated their operations, and reached the shallow groundwater through the infiltration. Next. Well, but deep wells that are upstream the tailings and the smelters also have very high concentrations of arsenic, as we saw. Next. And we didn't know what to who would be the source of this arsenic. We found also difference in the chemistry general of the shallow polluted wells and the deep wells, as you see in this figure, Schreller diagram. Next. And well, we identify the minerals in the area. As you see, there are many minerals, arsenic-bearing minerals, different that are present there. Next. And analyze samples, rock samples from that had some mineralization and the others from the same formation that didn't present, and also analyze the sulfur isotopes and relate with the temperature and made some geochemical modeling and identified that arsenopyrite oxidation and scorodite dissolution are releasing the arsenic to the deep groundwater wells. Next. Well, the water flows through fractures in this deep limestone fracture aquifer from the oxidized zones. Next. And has contaminated these deep wells that are used for us drinking water source. But some actions have been taken after our identification. Next. Like the build of an aqueduct, but the problem is that the well is far from Simapan town. It implies high energy costs and had faced political difficulties, and also has a low discharge. Next. Treatment plants that were installed in 2000 13. Next. But
3
they
minutes,
have
Maria.
faced the operation and maintenance difficulties. Next. Yes. Next. We also developed geology and batch and column experiments and found that limestones may also be used to retain arsenic. Next. And treat acid mine drainage with native limestone also was effective treatment. Next. Well, this is the column experiments. Next. Well, the next is in León area. Next, León City is a big city in Mexico that is a very important shoe making and leather factories. Next. And there we study the chromium presses. There's some uses of chromium, as you see there. Next. And we studied the sources and levels of chromium in groundwater that were first ascribed to the tanneries. And next. But we found that there was also a chromate factory implied. Next. And other sources. Well, these are some of the health effects of chromium, mainly hexavalent chromium is the toxic form, and its inhalation is the most big exposure, but it also produces problems in the stomach, liver, kidney, etc. Next. Well, chromium 6 is very water soluble, and chromium 3 is required for glucose metabolism, and it precipitates the pH, normal pH in groundwater. Next. So this is the EH/pH diagram of chromium. Next. We sampled in the valley, in all the valley wells, deep wells mainly. Next. Next, this is the wastewater. And well, these are the difference we measure between total chromium and hexavalent chromium in the water used for irrigation. Chromium 6 was not present, but in the groundwater southwest of León Valley, chromium 6 was reached very high concentration, up to 82 milligrams per liter. Next. Well, northwest of León Valley, there is the recharge zone. Next. And we find there, next, rocks with high concentration, chromium concentration, that ultramafic rocks that had over 4,000 milligrams per kilogram. Oxidation with CO2 and water, interaction with water produces high chromate concentrations that infiltrate to the groundwater in one of the areas. Next.
Okay, Maria, we're at time. We're over time right now. Can you wrap it up, or should we come back later?
Oh, well, we may come back later.
Okay,
No
all right. Thank you. Thank you very much. That's a ton of really awesome work done all over the place in Mexico. So I really appreciate it. We have to bring up our next speaker, Yana Asanta Navarro. So she has to take off right after this. She's doing a ton of experiments today, but she's blessed us with her presence and talking about my favorite study organism, looking at food quality influences on Daphnia salt tolerance. So this is sort of ties back to Sally's presentation, looking at ecological effects of salt.
Yeah, thank you, Clay. And like you said, Sally really did a nice job in introducing you all to the salt problematic already. Just before I get started, this work has been during my PhD, so with the University of Constance in Germany, in collaboration with Professor Shelley Arnott in Canada, Queen's University. I'm currently in the US. Next, please. You can actually go to slide 5 already, since that is all the reasons why we have salt in our water. No, one back, please. Back. That's, yeah, exactly. Like Sally already said, so I don't need to get into much more detail there. And the important message basically is that our freshwater ecosystems, lakes and streams are increasing in salinity. Next, please. So why is that important for our freshwater organisms? So we're basically looking in this study at Daphnia, that's the little animal in the middle, keystone herbivore, major food source of fish in lakes, and is basically preying on phytoplankton. Now, Daphnia are very important for the lake ecosystem itself, and Clay is going to talk a little bit more about Daphnia later on. But we basically used those Daphnia, and all organisms in the freshwater ecosystem are protected. Next, by water quality guidelines. In the US, the threshold is 230 milligram of chloride per liter. In the European Union, so all countries that are part of the European Union, 200 milligram per chloride per liter, and in Canada, it's 120 milligram chloride per liter. Next. So, like I said, this study was done with my amazing partners. You can see here on the right, Shelley Arnott, Tony Clauschies, and Dominic Martin Kreuzburg. And it's being conducted in Canada at the Dorset Environmental Science Center. Next. And what we've been wondering is how food quality, so basically what you have to imagine is that phytoplankton differs in their food quality. So depending on what the Daphnia eat, they might actually differ in their response to salt. Now, food quality, there's different parameters you can define as food quality: ingestibility, digestibility, toxins, elemental nutrients. What we were particularly interested in is biochemical nutrients. So first of all, sterols, you can see an example of cholesterol, and then polyunsaturated fatty acids, there's an example of EPA. Next. Now, those essential lipids are pretty important for physiology, for they are precursors for hormones, for example, so really important, and they're essential, so they need to be taken up by the food source. The Daphnia cannot synthesize them de novo, so out of nothing. One pretty important function of this essential lipids are also their role in the membrane. So what you can see here are basically the double bilayer structure of our eukaryotic membranes with the phospholipids there. And what you can see is when you have saturated fatty acids, basically having a straight line. So the packaging of the phospholipids is pretty tight. So this is decreasing fluidity and permeability, which is pretty important for salinity, of course. When unsaturated fatty acids are incorporated, and because of the double bond that they have in their fatty acid chain, you have a tilt. So basically what happens is the phospholipids will be pushed aside a bit. So you will have an increase of fluidity and permeability. Sterols have the opposite effect again, and you can see that they have been incorporated here in the little picture with saturated fatty acids, but you could imagine also that they would be incorporated when you have saturated fatty acids in the membrane. They stiffen the membrane, and they're also making it more dense, so they also decrease fluidity and permeability. Now, what would we expect if we expose an animal to sterols or to polyunsaturated fatty acids? Basically, it should be more susceptible because of an increased permeability in the membrane to salt when exposed to polyunsaturated fatty acids. And the opposite should be true if we're exposing them to sterols in their food, basically because the membrane would be packed denser and the permeability would be decreased. That's exactly what we did. Next slide, please. And the result you can see here. So in the blue line, we can basically see the LC50 value. So that's the lethal concentration at which 50% of the population were dead or alive, depending on how you want to look at that, without any supplementation in the food. And when we supplemented them sterols, the LC50 value increased to 138 milligram per liter of chloride, and when we supplemented them polyunsaturated fatty acids, it decreased to about 50 milligram per liter. So basically, by what they were eating, their food source kind of determined how resistant they were to salt contamination, or which concentration they could still tolerate. Next. And so this is coming in line with two other studies that are basically claiming the same. That's also the claim we are making in our publication that you're welcome to look up. Shelley Arnott, lead author on the one on the right, is basically saying that there is a species-specific dependence of chloride resistance or tolerance depending on which Daphnia species you're looking at. So we have a species-specific relation between chloride tolerance and the other species. Then Norman Yan and his colleague Brown in 2015, the lower graph, looked at food quantity. And if you look at the x-axis, that's basically going from a very oligotrophic, so 0.2 lake, to a more eutrophic lake, so more nutrient-rich, 1 milligram carbon per liter. So when we're looking at the 0.2 one, let's assume a very nutrient-poor and oligotrophic lake, we would basically see that if you're feeding them little food, so they have low food availabilities, you would have only an LC50 of 50 milligram per chloride per liter. So they can only tolerate 50 milligram chloride per liter in an oligotrophic, so in a nutrient-poor, very clean lake. And when we're looking again at our results, basically when we're not giving them sterols, and when is that the case? So cyanobacteria, for example, not eukaryotic, but actually bacteria do not contain any sterols. So when you're assuming a lake, for example, where there's lots of cyanobacteria, you would assume a reduction in sterol availability. So then we would have a reduction of the salt tolerance to about 80. And if there's, on top of everything, lots of PUFAs available, then it might be reduced to 50. So 50 milligram chloride per liter, that's what we found. Brown and Yan, a similar concentration. And next, please. If we come back to the thresholds that those countries have, and especially the the countries in the northern hemisphere, of course, very subject to this road salt contamination, or in general salt contamination into our freshwater ecosystems, then we can see that 50 milligram carbon chloride per liter, what we found in our study and also what our colleagues found, is way below of our current thresholds. Canada is doing the best job so far, but even there, water quality guidelines across the northern hemisphere really need to be adapted. And what all authors of all three studies that I showed on the previous slide are really agreeing on is that we need site-specific water quality guidelines. So for example, taking into account the trophic state, so the nutrient load of a lake would be a good starting point, for example. And that is it already. Thank you for your attention.
Excellent. Thanks, Jana. Good luck finishing up today.
Thank you. Have a good day.
Okay. So next up we have me, and thanks really to Sally and Jana for sort of setting up my talk. So today I'll go ahead and set my timer so I can cut myself off if I go over time. So today I'll be talking about some work that I did in association with my last postdoc, looking at working in southwestern Greenland, looking at the effects of geochemistry and salt on biological elemental variation. Next slide, please. All right, so as a biologist, this quote really from the late great R.J.P. Williams really, I think, kind of summarizes my interest in biology. And I'm really interested on how elemental availability in the environment influences biological organisms. So influences how they function, how they work, and sort of interactions between organisms and the environment. So we know that sort of this elemental availability has really important ecological consequences, such as this figure in the top right here, where we know that sort of changes in redox chemistry in oceans sort of altered elemental diversity in the marine systems through time. And we know that there's a pretty good correlation, for instance, with redox sensitive elements and diversification in ecological systems. So there's ecological effects. There's also evolutionary effects. For instance, when superorders of algae in the oceans evolved under more reducing conditions, we see pretty big differences in their elemental composition versus sort of newer evolved superorders. So basically, we can tell a lot about ecology and evolutions and organisms interactions in the environment by looking at their elemental profile. So that's what I'm really interested in doing. Next, please. And I got the really unique opportunity to do this for about 3 years, playing around in southwestern Greenland. And there's all kinds of really interesting gradients. I could talk about them all day, but for the purposes of this talk, there's really big salt gradients. So basically, as you go from right to left across the screen, so that arrow is just showing there's really big differences in salinity across the region, and increasing as you go away from the ice sheet. In fact, we actually get some some oligosaline lakes, which is that yellow point on that map. So I'm not going to talk as much about salinity today because it's already been covered. I'm interested in talking a little bit more about the effects of variation and geochemistry on biological organisms in the system. So basically just went out to all these lakes, collected water, collected microbes, and collected our cute little Daphnia up here. Okay, so next slide, please. And I can't really super get into all the elements on this. We can talk about it more later. But basically, this is just a PCA showing differences in the elemental proportions across these lakes, where you can see I've color coded the lakes in different colors, and then I've just sort of drawn circles around lakes that are similar to one another. So basically you can see that sort of starting out at the fjord, the lakes in blue, they're quite separate from the lakes of the ice sheet. And we see this not in just the geochemistry, but also a lot of physical conditions across these lakes. So they're quite different in those two different regions. And we see that although the saline lakes have a ton more ions in them than these other lakes, when you're just looking at relative proportions, they're similar to the ice sheet lakes. Next slide, please. All right, so how does this affect the biological organisms? Well, we see that the microbes, or as I like to think of them, Daphnia food, also show really similar patterns in their elemental composition and how they sort of break out across these regions. So again, we see a pretty distinct differences in the fjordal lakes from the ice sheet lakes, and a little bit more overlap between the saline lakes, which is kind of interesting. But basically, we also see a little bit greater overlap just in general in these microbial organisms. And of course, that's because biological organisms have a relatively more fixed requirement of elements than they're supplying the environment. So there's going to naturally be a little bit more overlap. Next slide, please. And Daphnia, actually their variation is even smaller, like I just said. So so basically the elemental variation is sort of going down as you go up the food web, but you can see these Daphnia are actually really distinct in their populations. So basically these fjordal lakes have lower concentrations of basically all elements besides maybe one, really high elemental concentrations of elements that we would normally associate with osmotic stress, and some larger concentrations in the ice sheet lakes of things like phosphorus and metals. So we think, again, remembering back to the introduction, this could suggest that these Daphnia are sort of adapted to these different populations. Next slide, please. And we're really interested in looking at that. So right now I'm currently working on looking at population genetic differences across these populations to get an idea of evolutionary adaptation. Love to follow this up with some creative ecological and evolutionary studies. And one of the reasons I invited some of these SALT people secretly was because I sort of like to start doing some of this work a little bit closer to home, perhaps even in research coordination network. So if you'd like to if you're interested in that, please let me know. We're sort of kicking around ideas right now. And finally, sort of in the discussion, if we could talk a little bit more about sort of ways of looking at these different elemental profiles kind of statistically is something I'm sort of interested in on the side. All right, I'm over by 20 seconds, so hopefully that's not too bad. So next up, I'm really quite proud and pleased to introduce our next speaker, Justin Edel. So he's an undergrad researcher. So he did an REU this summer up in the Experimental Science Station in Wichita. Do we have his presentation? Yeah, so so Justin did a ton of work this last summer, basically all summer long, looking at the effects of drying, rewetting, and vertebrates on wetland ecosystems. And this is a really big study with a lot of people that were doing a lot of different things, but he's going to get us some experience giving a presentation today. So Justin, are you online? Are you ready?
I'm here.
Awesome. Take it away.
Thank you so much, Clay. I was really fortunate to get to be involved with this project over the summer. So we just get right into it. Can you go to the next slide? So why are we worried about dry and re-wet? Well, it has an important influencing factor over the ecology of our and connectivity and biogeochemistry of our stream systems here. And the importance of intermittent streams is that the majority of global streams are intermittent. And they're showing an increasing prevalence with our global global climate change. And then here in Kansas, 60% of the streams are actually intermittent, and they make up a network of isolated and connected pools. Next, please. So the effect of aquatic vertebrates on these systems depends pretty much on their presence. So they can sometimes be extirpated after drying events. Fish can colonize or recolonize separate pools after refilling events where the streams connect the pools. And then amphibians can colonize just over land because they're more mobile over land. Next. So one of our first hypotheses is that drying will affect the nutrient composition in these systems. So we had a mix of drying and trophic treatments for this experiment. So our drying treatments were a non-dry, where these tanks were filled up and were not allowed to dry, and then a dry re-wet, when they were dried down completely, left for a period of time, and then refilled when the experiment was ready to start. Can you go to the next slide, please? Our hypothesis 2 is that the presence of vertebrates will also affect the nutrient composition. So our trophic treatments were a no vertebrate control, a treatments with larval amphibians, some with fish, and some with larval amphibians and fish. Next slide, please. Thirdly, we wanted to we were saying that the effects of drying and vertebrates will also change over time. Next slide. Next. So on our sampling days, we would go and collect samples from each tank. This is my daughter and son. They helped me with some of the sampling. And some of the analysis that's relevant to this talk would be, we did an ICP OES, which is what our results are looking at today. Next. Next, please. So we were able to do this sampling over a period of 12 weeks, and we had a total of 17 sampling days. And so our preliminary data that we're looking at today covers day 1, 12, and 33. And it might be important to note that the peak metamorphosis actually occurred after these days at around day 40 or 47. Next, please. So this is a PCA that's showing the differences between the geochemical differences in elemental concentrations between our dried and undried treatments. So on the left in blue, you can see our undried treatments there, or sorry, our dried treatments. And then on the right side in the green, we have our undried treatments. So this is the majority of variations associated with drying on the left and right. And you can see that our undried actually had a higher concentration of elements. But you could also see in that yellow oval there that you have an overlap with our dried treatments. And to really see why that is, you kind of have to see the effects over time. Next. So this is the same PCA, just showing effects over time from day 1, 12, and 33, with 33 up in the right in the green there. And you can see these elements concentrations change. So you see a shift from more potentially toxic elements to a grouping of more essential biological elements. So, and then by day 33, you see that these drying effects are greatly reduced, which would explain our overlap that we have between the dried and the undried. But what is overriding these drying effects? Next slide, please. So we need to find that out, we need to look at our vertebrate treatments. So in this PCA, this is a PCA of day 33, showing our vertebrate control and control treatments. You can still see that there is a drying effect. If you look at the y-axis, we have a separation of our dried on the top and our non-dry on the bottom. But the majority of the elemental variation in this graph comes from the vertebrate treatments versus the control. So we have our vertebrate treatments on the right in the blue and a control on the left. So as you can see, the drying actually does have quite a large effect, but when you add a vertebrate colonization, it definitively alters the nutrient cycling to a pretty great effect. Next. And so what do we come away with? So far, these are all still preliminary. We haven't analyzed everything yet, but we see that the elemental composition of the water is initially different following a refill compared to the pools that had not dried. However, those differences get caused by the drying lessen over time, and differences instead become dominated by the presence of vertebrates. Interestingly enough, the identity of the vertebrates did not appear to have caused any large differences. Next, please. And for the future, with these are preliminary data, so we intend to look at them more closely along the timeline and understand the smaller scale and temporal patterns, and then focus on a smaller subset of especially important elements. And next. And I just want to acknowledge our Mine Clay fundings for this, and thank you very much for having me.
Awesome. Great job, Justin. I think you did better than I did for sure. Yeah, really cool study that I was super excited to be affiliated with. Does anyone have any questions for Justin?
No.
He did a really good job presenting it, yeah?
Yes. All.
Thank you.
Everybody.
Okay. Does anyone have any questions for any of the other speakers in this session? So Yana and Sally had to duck out. Does anyone have any questions for, I guess, I think Maria had to leave as well, for Nicole or I about what we're doing? So it's a little bit different. I mean, mine was just basically like stuff I'm interested in. I don't know that it actually fits the remit, but I've been doing a lot of work with Michael, looking at these elements and all these different elements. And I just love these kind of formats because I haven't had a lot of proper training in geochemistry. So I learned a lot about it. I'm trying to learn by myself, but I think I'm really interested, for instance, in using ICP-MS and stuff. It generates so many elemental signatures, and as biologists, we're usually just interested in maybe, or as biogeochemistry, we're looking at like a handful of elements, right? Whereas a lot of the work that we're doing, it seems like there's all kinds of not even biologically essential elements that are cycling through the environment. So Sally showed, I think, pretty good evidence of salt, but I think that there's so much work that we could be done sort of working together, and then people with, different backgrounds. So, Ophelia, for instance, and Maria, looking at toxic chemicals and sort of bringing those into, like, we can learn a lot biologically from interacting. And that's why I'm excited sort of working with the SEGH now and going forward. So I think to close the session, Michael had sort of suggested we might discuss just a little bit, if everybody could kind of hang on, about sort of just maybe what the future of the SEGH might be. There's also some people I know, maybe they're still in attendance, who suggested that they might like to join SEGH. So we can talk about that a little bit. Yeah, Ophelia?
Yes, I am very happy because it was it was a small one, but it was very deep, very deep. But I think we have to do, this time is difficult with our students, but I think we have to do more. We have to try to include our students in these kinds of works.
Yeah, that's good. And I think we definitely have place for students to be involved. And yeah, I mean, so this was really driven actually more by, Alistair, who had his stuff together more than I did, and also Haley. They did a lot of the initial work on this to put it together. So they have a lot of energy for doing that. And I'm all for, getting people involved. It's been really hard, pandemic stuff, and getting interest, and honestly, just the drive to kind of do stuff isn't there. But
I think you've done the first one. That's the most difficult one, yeah. And it's going to be easier now.
Yeah,
and
sure.
Hopefully other people will be interested in helping you out. And maybe this is the first of many. I do like this short format.
Yeah,
it's quite easy to access.
It's not a bad way to spend an afternoon. Thanks for sticking in there, Michael. You must be tired.
That's right. No, it's wonderful talks. Thanks to everyone and the effort for putting this in, because it does take time to think about doing this.
Yeah, thanks.
Maybe you could think of prizes, whether you do it for this one or the future, Clay.
Yeah, well, I mean, like that's why I was saying about memberships. If there's anybody interested in joining and would like to kind of help grow the society, especially students and stuff like that. Everybody has my email, so maybe contact me. And there's definitely a few people that said they were interested in joining on the sign-in sheet. So I'll definitely be bothering you via email. And I think also
I put a link in the chat.
Off, I think Alistair posted a link to kind of how to join SEGH and get signed up. So
it's a fantastic opportunity. It's a fantastic opportunity to grow from a low point. With the Americas was a very key. Obviously, you heard of Bob's history, but it's an opportunity to get in at the beginning and try and regrow the Americas rather than a USA focus, perhaps a bit wider.
Yeah.
We've certainly seen this kind of energy in Africa from early career researchers who have really got some energy and creating a network amongst themselves. And a great experience for doing that. So yeah, I'd definitely encourage other involvement. And thank you, Alistair, for doing this, because you've also just been through putting your thesis in as well. So
yeah.
Oh, it was these presentations were so great, and I'm so glad to be a part of it.
Dr. Alistair, excellent.
Well,
plus,
I'd like to.
Plus, that having this line on your CV, that's going to land you basically whatever job you want. It's pretty good street cred. So
that's not why I did it. It was so great to work with you, Clay, and Bob, and everyone else in the Americas section to put this together. And I'd also like to echo thank you to all the speakers today. It was really fascinating to hear about research that was so removed from my own and yet still related in ways. So yeah, thank you to everyone.
Yeah, that's one thing I love about it. It's like anybody that measures anything can kind of come and put their spin on it, and that's what's really fun. And yeah, I definitely enjoy all of it. So yeah, we'll do this again and try to keep going forward. But yeah, everybody remember that Bob had made an announcement, and as far as pushing early career stuff, I think his is more targeted at undergraduates, right, Bob? Or is it high school students? I can't remember. I think it's undergraduates.
You've blocked my video. I know I'm not that good looking, but I can't.
There we go.
Yes, it is. We welcome any students at any level, undergraduate, graduate. We'd even consider a postdoc, early postdoc. So we welcome anyone and everyone. Again, to repeat, there are perhaps a few people that joined after the initial introduction. SEGH, along with half a dozen other related organizations, are sponsoring a student-oriented virtual conference on environmental geochemistry / medical geology. We're doing this on a regional basis. So the first of these conferences will focus on Latin American issues. And it will be October 18th and 19th. Deadline for abstracts appears to be the 25th, but we'll accept abstracts through the end of the month, certainly. The next such conference, we're thinking of Africa. So hopefully this coming conference will be as successful as this one, and we'll run a series of conferences. Let me throw out an idea that occurred to me listening to all of these presentations, that perhaps in 2022, we could have a theme-oriented session talking about how climate change is impacting our research, whether it's enhancing it, diminishing it, altering it. But a lot of what I heard this afternoon it occurred to me that climate change could have substantial impacts on what the researchers are doing. So it's just a thought I want to throw out, but I also want to commend you and Alistair for doing a great job. I thought this went exceptionally well, well planned, well executed. Even though you gave yourself an extra 20 seconds,
Yeah, I know. I'm sorry.
we'll forgive you that. You earned it.
Yeah.
20, maybe not 30 seconds, 20 seconds.
Okay. Thank you. Yeah. Okay. Does anyone have anything else to add? I really am thankful. Malcolm, thanks for showing up and kicking it off. That, like I said, that was a good crash course. I really appreciated it. Yeah, and thanks to Alekos. So he sent this out to a lot of his colleagues, and he definitely drove some eyeballs here. So I really appreciate that. Yeah, and thanks for everybody for joining. We definitely had, you know, people from yeah, Europe, Africa, South America, North America here. So it's pretty good. I don't know if we got anyone from Asia, but yeah, really proud of this first attempt, and we'll be bringing another one back for the Americas. And I'm sure that Michael, is there another one planned for Africa or Europe?
There's a plan for something in Asia, so
okay.
Led from Pakistan, so that could be happening over the next month.
Okay.
I'm toying with something one side of Christmas. I'm not sure quite yet, partly for the handover of the presidency to successor, so that might be a quick meeting. We certainly have a cohort of 50 African early career researchers for a writing workshop, so we may build up to them providing some presentations in the new year.
Yeah, that'd be really awesome. I'd love to see that. So
yeah, and we may even we've got plenty of French speakers, some Francophone Africa, so maybe we'll have some alternative languages.
Sweet.
See how Google Translate works. Eh?
That'd be fun. Yeah, I like it. Okay, well, I've got to run to school or bike to school and fill out some forms. But yeah, thanks everybody for attending and presenting. I really appreciated it. And I'll do some follow-ups on email afterwards for anyone interested in joining and stuff like that. So thanks, everybody. Have a good rest of your week and a good weekend.
Okay, cheers.
Thank you, everyone.
Thank you.
Bye.
Bye. All right, thanks, Alistair. Go celebrate. Yeah.
Thanks, Clay. The recording you'll put up maybe on the website or something as well?
Yeah, for sure. Yeah.